All right, looks like we are live. Welcome everybody to Standing for Truth. I am your host, Donny, and tonight we have another episode of Late Night with SFT. It's actually been quite a while since we've done one of these later shows and streams, and so Jackson and I, we thought, you know what, since we're debating a few issues and debating a few ancestry, creation, evolution related topics over Facebook, we thought, why not bring it into an open mic rather than spend all night arguing over a keyboard. So, Jackson, I've always complimented you on the fact that I appreciate you're not a one of them keyboard warriors. So you're willing to engage the data, engage the ancestry discussion. So Jackson Roe, after show, what's going on? How you been? And what's up?
I'm doing good. Supposed to have a date tonight. That's why I'm up so late. But she's coming Friday now, apparently. Girls are weird. But yeah, that's about it. Man, I'm just supposed to do this like 2 hours ago. Been waiting all night.
It's been a busy day. It's been a busy day, which means this is probably not going to be, you know, the typical 6-7 hour open mic after show as Logical, Plausible, Probable does. But we'll have some fun for a couple hours with the new baby. My sometimes all nighter schedule. It doesn't work too well for me anymore. So we'll do this for a couple hours. Should be fun. Jackson and I will start off going over a few topics. I think specifically ERVs, a little bit on human evolution, just separate ancestry in general, right? Universal or separate ancestry. Bubblegum Gun says, Jackson, get yourself a sweet Lambo poster. So appreciate that, Bubblegum Gun. You know what, Jackson? Your evolution in terms of your audio and webcam since you first started doing these debates, I would say is pretty good evidence of large scale evolution, because you've gone from like, you know, hardcore low def, webcam to HD quality, solid mic, no lag. So there you go.
Yeah, proof of evolution right there.
Proof of evolution right there. So, all right, why don't we get right into the discussion? Actually, first, I'll kind of plug our creationist clothing. I just got a bunch of new SFT, Team Standing for Truth, creationist clothing in. This is the heterozygous Adam and Eve shirt. I've also got our SFT logo shirt, and just various other Standing for Truth Ministries related shirts, guys. So please check out creationistclothing.com, because we've got mugs. I've got SFT mug here.
You should make SFT globes to piss off the flat earthers.
Yeah, right there. I'll get the SFT logo right up on that globe in the back. So yeah, if you guys like kind of the SFT gear, check it out.
The Mormons beat you to the creationist clothing. I think they got the Mormon underwear.
Yeah, the Mormon specific underwear. So all right, well, why don't we kind of get right into the first topic before we end up opening it up here. And I was going to share screen because we were kind of talking about endogenous retroviruses specifically. And we were discussing this paper and the findings there. I've got a couple portions of it highlighted. Long disregarded as junk DNA or genomic dark matter, endogenous retroviruses have turned out to represent important components of the antiviral immune system. You know what, Jackson?
Yeah.
From your position, okay, somebody who holds to universal common ancestry, somebody who obviously holds to ape-to-man evolution, and would say that the chimpanzee is our closest cousin or relative. Why don't you take a minute or two to explain ERVs from your position, since a lot of evolutionists think this is one of the, if not the best evidence for evolution. Take a minute or two. What are endogenous retroviruses, and why do you believe they provide evidence for evolution? And then we'll engage it from there.
All right. Basically, I think we have different interpretations of how endogenous retroviruses came to be. Like, generally, it's thought that viruses infected gamete cells, and it gets kind of spliced into the genome and passed on to the next generation. But I think you think something different. I forget exactly the details. I don't want to misrepresent it. But there are 98,000 ERVs in the human genome, which, according to most sources, is more than genes in the genome. So I don't doubt that some of them have a function, but not all of them do. And I was looking into this earlier. Some of them have a negative function. Some of them are associated with cancers and even autoimmune diseases. So we share 99.8% of them with chimps. And they can kind of infer the age of certain ERVs by the number of mutations acquired per ERV, deactivating mutations, and the long terminal repeat sequences, which are vary in length from like 300 to 500 base pairs long. So that's pretty much what ERVs are, and they're 5 to 8% of the whole genome, so they make it a huge part of it. So that's what I got on ERVs right now.
All right, I appreciate that Jackson. So, in a nutshell, from the evolutionist starting point or perspective, these endogenous retroviruses and, of course, other classes of retrotransposons, the reason why we share so many, right? And we can even talk about ALU sequences. The reason why we share so many, according to the evolution model, an ape-to-man evolution between, let's say, humans, chimpanzees, bonobos, gorillas, all the way down the line, right? All the primates, essentially. And why there's a pattern of like nested hierarchical patterns in terms of the placement of these so-called insertions, right? Is because we've inherited those and we've passed those on because of millions of years worth of ancient viral infections. So millions of years ago, we had these ancient viral infections, obviously, that have inserted themselves in the genome, and they've been passed down ever since. And obviously, there's been splits, right? So when the human and chimpanzee line, let's say, split from their hypothetical common ancestor, they would have taken with them these ancient viral remnants. And this is just observation today in the present of shared relationship between, let's say, the primates. Would you say that that's kind of an accurate representation?
Yeah, that sounds about right.
Okay. So, and I've said this many times. I think the question comes down to, when it comes to the creation and evolution model, or separate ancestry or independent origins, we would say that humans are independently created from any other form of life, right? Humans share no ancestry with, let's say, the great apes and everything else down the line. So the question would be, are these really the ancient remnants of past viral infections millions of years ago that have been passed down? Or are they created units of DNA function? So our issue and point of disagreement was you pointing out from your position or your understanding that these findings, that these endogenous retroviruses are functional in so many different ways, right? This is just one paper. I mean, they're functional in determining cell types. They're functional in regulating genes, functional in the embryo. They help with cell stress process, like pseudogenes. They're essentially necessary to sustain healthy life processes in the cell, right? You don't see how these findings would be contradictory to your model of evolution, essentially, right?
No, I think some of the material could have been co-opted into it. I don't think it's necessarily a problem.
Would you say, from your understanding, did the evolutionary community predict function? This is an important question, actually. Did the evolutionary community predict function before function was found?
Well, there's been a lot of junk DNA that's turned out to be functional, so I would say they probably got that one wrong. But I'm not 100% sure.
Okay, so would you be pretty comfortable saying that the evolutionary community probably did not predict functionality for these endogenous retroviruses?
They may not have, but I mean, there's pretty much no way they all do something, and some of them physically can't with all the deactivating mutations they have.
Okay.
But I don't doubt that a lot of them do.
Okay. So do you not see how that would be a little bit post hoc, ad hoc rationalization? The whole co-option counter argument to the function associated with endogenous retroviruses. What are your thoughts on that? Do you think it's a little post hoc, or?
Sure, A little bit, but it does appear to be viral genomes basically spliced into the thing. It doesn't appear to be units of created function. Like they appear to be gamma retroviruses, epsilon retroviruses, beta retroviruses, and delta retroviruses. So I'm not sure why they would be created to look like viruses if they were just units of functional DNA or a gene or something like that.
Right. Nobody's saying that they don't look like viruses or they have viral properties or characteristics and traits, right? Mm-hmm. Viral-like elements and just viruses in general. We've discussed, Jackson, in previous shows and debates, how we have over 10 trillion cells. And yet we have more, for one, bacteria than we do cells. But we have more viruses in and on our bodies than we do cells and bacteria. Well, if viruses are bad and always harmful, like people seem to have this assumption when it comes to viruses, then why aren't we dead, right? How are we able to survive with this amount of viruses in and on our body? Well, it's because most viruses are good. They're beneficial for the ecosystem. They're beneficial for all kinds of species and forms of life. They're beneficial to us, especially in terms of regulating the number of bacterial species in our body, right? The virome is incredibly important. And if we didn't have viruses in and on our body, then guess what? We'd probably die just because of the number of, let's say, bacterial species in and on our bodies that are not being regulated or managed. So we have massive numbers of viral-like elements, endogenous retroviruses being one of them. And I like this in the paper right here. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. Notice that, regulators of antiviral gene expression, helping us to pretty well fight the negative effects of bad viruses. Not only regulate cellular immune activation, but may even directly target invading viral pathogens. These ERVs there, okay, directly targeting invading viral pathogens. I mean, that's an amazing benefit and function. Last thing I'll say, and then I want you to take plenty of time to respond and give your thoughts. This is from an article on Evolution News. Junk, no longer junk. ERVs are integral and important components of immune responses. ERVs frequently have important immune functions, and they should not be presumed to be junk DNA. This defeats both the junk DNA argument against the design of the genome, human and otherwise. It also challenges those who want to use the supposed junk status of ERVs as an argument for common ancestry. After all, if ERVs have functions, then, and here's the important part, because evolutionists want to say shared endogenous retroviral sequences are evidence for common ancestry. Well, wait a minute. Then shared ERV sequences in similar locations across the genomes of different species may reflect functional requirements rather than mere common ancestry. So humans and other species are designed with trillions and trillions of viruses to help regulate aspects of cellular processes, regulating the number of bacterial species that we have, and viral-like elements, which there's a hypothesis called the VIGE hypothesis, variation-inducing genetic element hypothesis, where it's proposed that these viral-like elements, including these jumping genes that can jump around the genome, turning genes on and off, inducing variation, for example, ALU sequences are, we now know that they're functional and necessary for various types of processes in the brain architecture. Okay, so these are there for functional purposes. So of course, we're going to expect to share then plenty of these viral-like elements and these various classes of retrotransposons with the chimpanzee and all other forms of life. I mean, it's not us. It's not like the ERVs name themselves ERVs. Sure, they're called endogenous retroviruses. And because people hear the word or see the word virus, they automatically assume, okay, something bad, something that must have inserted itself millions of years ago and it's been passed down. Okay, so no, this makes sense that the ERVs would have viral-like characteristics and traits because there's a variation in the type of viral-like elements that exist, right? So I said a lot there. What are your thoughts, Jackson? Essentially, it comes down to this. And for the audience, it's as simple as this. We've always predicted that ERVs should be functional. We have now discovered that they are functional, which means ERVs are evidence for common design and not common ancestry. Now, if we found that the ERVs were junk, useless, and a reflection of evolutionary history, right, common descent, because they're pretty much useless evolutionary leftovers, genomic fossils, then yes, they would be evidence for common ancestry. But that's not what we find. Jackson, go ahead. Take your time and try and just, I guess, critique all that. What are your thoughts on that?
Well, basically, I'm not refuting that a lot of them have function and positive function, but some of them appear to have negative effects, like causing cancer and autoimmune diseases as well. But out of 98,000 ERVs, it's not likely they all are critical. And there are several unique to humans and a few unique to chimps. And the further back you go, there are more unique to gorillas, even more unique to orangutans. So to say that they're all necessary for the same reason across species doesn't really make any sense, because then all the species would have the same number of retroviruses, wouldn't they? Endogenous retroviruses. But it creates this kind of tree of what appears to be inherited ERVs.
Good points, interesting points. And first thing I want to say is we have not just discovered 1 or 2 or 10 functional ERVs. No, we've found massive numbers of functional ERVs. We've discovered functions for pretty well all types and all classes of retrotransposons. For example, I pointed out earlier, even ALU sequences. Intronic ALUs influence alternative splicing. Our results indicate the importance of intronic ALUs in influencing the splicing of flanking exons, further emphasizing the role of ALUs in shaping of the human transcriptome. Okay. ERVs. Scientists identify new beneficial function of endogenous retroviruses in immune response. Endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. So based on that, I want to ask you a question in 1 second, but I do want to address everything. Okay. Far from being junk DNA, the pervasive retrotransposons that populate the genome, notice this, the pervasive retrotransposons that populate the genome have a powerful capacity to influence genes in chromatin. Okay, a new study demonstrates how the transcription of one such element, HERVH, can modify the higher order 3D structure. I mean, this is amazing stuff of chromatin during early primate development. And I mean, we can go on and on and on. Retroviral promoters in the human genome. These data suggest that ERVs may regulate human transcription on a large scale. Okay, so just like anything, one single point mutation on, let's say, a healthy gene can result in death, can result in disease.
Sure.
Okay. So endogenous retroviruses and these various types of retrotransposons that populate the genome of living organisms can also be subject to what, Jackson? Mutation.
Yeah.
Degradation. And what can that lead to? Disease, cancer. Okay. A once functional, let's say, viral-like element that's beneficial and necessary to sustain healthy life processes that's hit with a deleterious mutation can, yes, result in disease. But that doesn't mean that the viral-like element or whatever DNA element you want to look to, let's say, you know, these number of pseudogenes, that doesn't mean that they started off bad or that they were designed bad. Just like that single point mutation that killed a person due to disease, it's not like that specific gene or whatever the location in the genome that was hit with a detrimental mutation was originally that way. Does that make sense? Yeah. And what are your thoughts on that before we move on, just so we hit every point?
That kind of seems like a post hoc explanation too. I think we're both kind of doing that a little bit.
But think about this. You can do mutagenesis experiments where, and they discovered this, that many portions of the non-coding regions, okay, because we've discovered that the non-coding regions of our genome act like a massive control panel that are turning genes on and off. You've got millions of switches, you've got gene regulation, essentially like a computer, okay? Yeah. And a lot of the non-coding regions of our genome are dictating the developmental process, okay? Deciding and determining how structures of the body are sequentially put into place and dictating scenarios in the various developmental windows. Okay, so there's just so much function going on. It's science fiction on a molecular level, essentially. Okay, so the point I'm trying to make is they've done experiments, and we've actually made predictions too, that if you mutate, okay, certain sections of the genome that, let's say, are assumed to be junk or useless, you mutate them, it's going to result in disease or cancer. That's because it's a functional section of our genome. And the reason why mutating it results in disease or cancer is because it's functional. I mean, that's just, it's not post hoc ad hoc. It's just reality.
Okay.
Go ahead. I said a lot there. What are your thoughts on that?
Well, if you're saying like the insertion of an ERV would be potentially fatal, you might be right. But in that case, the offspring would probably just die and that wouldn't be passed on.
Well, I'm saying if a position in the genome, let's say a functional DNA element in the genome is hit with a mutation.
Yeah.
Okay. That can lead to disease is yeah. What I'm saying. And here's the thing, we're going to expect it's not like we're going to expect the exact same amount of, let's say, endogenous retroviral-like elements in the chimpanzee, the exact same in the gorilla, the exact same number in the bonobo, the old world monkey, the lemur, the orangutan, especially, okay? Because there's differences in physiology, morphology, anatomy. There's differences in the phenotype of the organism, which means the genotype and the genetic content is going to be different depending on what creature you're looking at. It's not like it's just all going to be the same. I mean, the phenotype is the reflection of a genotype, right? So why would we expect the genetic content of these various creatures to be exactly the same exact number of endogenous retroviruses, the same exact number of allele sequences? I mean, Jackson, consider this. Even the similar sequences between humans and chimpanzees have massive differences when it comes to epigenetic modification and gene expression. So similar sequence, but massive differences in these epigenetic tags, the way certain genes are turned on and off, massive differences in gene expression in similar sequences. That's because the creatures are different. It's not like we're going to expect them all to be exactly the same. So what are your thoughts on that, Jackson?
Sure, they're not going to be exactly the same no matter what, but just the fact that it does form this tree, it just seems very coincidental.
Well, what do you mean by it forms a tree? Because,
well, tracing it backwards through, chimps have more in common with us, gorillas slightly less, orangutans slightly less, old world monkeys less.
But we still have specific endogenous retroviral sequences in humans that don't exist in chimpanzees.
Sure, a few of them.
Taxonomically restricted ERVs.
A few, yeah. I think it's like
it can be a few or many.
Yeah.
It's still taxonomically restricted to humans. So how did those come about in the human line?
They would have been inserted after the human chimpanzee split about 6 to 13 million years ago, I think it was.
Is that just a guess? Was this observed?
Well, of course it wasn't
inference?
Observed.
Right. Okay. No, yeah. So this is inferred.
Right.
Okay. So here's the problem. Here's the big problem. And here's where I think the fatal blow exists for the common ancestry model. Okay. Is since this prediction of function when it comes to endogenous retroviruses, we'll just stick with that for now. These critical functions in the embryo, determining cell types, okay, functional in the immune system, cell stress responses, so on and so forth. If you're saying that the ERV, okay, that this is evidence of an insertion, and since we have taxonomically restricted endogenous retroviral-like sequences as well, that means, there had to have been an insertion sometime after the split. Well, now that we know that these ERVs, okay, there's these important functions associated with these ERVs, now you have to explain not only the insertion, why there are taxonomically restricted endogenous retroviruses, but you also have to explain the function. So how long after the insertion, okay, and then these sequences were passed on to reflect ancestry, apparently, how long after that did these functions come about? Was it right away? Was it a couple 100,000 years later? Go ahead.
Good question. I'm not sure if there's any way to know that. I sure don't know it.
Okay, so let's even assume, for sake of argument, that there was an insertion and it was passed on. Okay, and that's why we have the taxonomically restricted ERVs. Let's assume that for sake of argument. And then let's assume that, a couple 100,000 years after that, a function is co-opted. This goes back to my original question that I've been asking evolutionists for years now. Show us the empirical data. Show us a non-functional endogenous retrovirus going from non-functional to something, not with just a minor function, but something incredibly functional, critically functional in the embryo. I mean, how do you explain that co-option? Maybe expand on that a little, engage that a little bit, Jackson.
Well, you're not going to like my answer, but it basically takes a long time to see something like that come about. And I know that's just a kind of a time of the gaps argument, but that's basically.
But here's the thing, though. It's not like you're just saying, it takes a lot of time to see, a one kind of creature to evolve into a drastically different kind of creature, right? And I understand the law of monophyly. We never outgrow our ancestry, right? Eukaryotes always going to be eukaryote. Mammals always going to be a mammal. Descent with modification, right? We're not outgrowing our ancestry, but we're adding new traits to that ancestry. Okay, so it takes a lot of time to get a single cell like organism to a whale, let's say. But again, you're invoking time to explain functional ERVs. But wait a minute, today in the lab, empirically, why can't we observe these non-functional ERVs? Why can't we observe the co-option of these incredible functions? That's where you can't just say, well, it takes too much time. No, we got to see empirical data that's even possible for a non-functional ERV. And this is why I like this slide. I just want to point this one out. If viral DNA is inserted at random into the genome, okay, which is what the evolutionary community would say pertaining to ERVs, it is much more likely to disrupt existing genes. A blindfolded painter will most likely mess up the painting he is working on by just applying random brushstrokes to it. But you're saying viral DNA is inserted, it's passed on, okay? But then at some point, it co-ops function in the embryo. I mean, expand on that a little, engage that a little. How's that possible? Well, we have any empirical data? Go ahead, Jackson. You can even rant. I'll give you permission to rant.
Okay. Well, it won't be a rant. It'll just be if it's negative to the embryo, it's just going to kill the embryo when it gets inserted into the genome. It's probably going to go no further. So it probably takes quite a few before you get one that's actually passed on.
But wait, but you're saying it's passed on. Mm. but you also have to explain the function. Notice a blindfolded painter will most likely mess up the painting he is working on by just applying random brushstrokes to it. So this viral DNA is inserted, passed on if it's passed on, but then these incredible functions have to also be co-opted or added to the ERV. I mean, that's where I believe it's fairy tale. That's where we leave science and we go to imagination.
I would talk about de novo mutations, but that's a whole other can of worms. So
here's the creation model then, because we've been talking very technical this whole time. So yeah. A lot of people in the chat, I don't want them to be confused. Okay. So again, it goes back to this question. What we're seeing, are these really the ancient remnants of past viral infections millions of years ago? Or are they created units of DNA function? We would predict function, and that's why we share them with various forms of life, because they were designed that way. They were front-loaded. Okay, so here's the creation model. It is possible to reinterpret retroviruses according to the creation theory. Accordingly, retroviruses could have originated from within the host genome instead of invading it from the outside. Okay, now here's the really, really powerful aspect of this model, Jackson. And I want you to engage this slightly. Okay. This had to have been the case in any scenario, since viruses are dependent upon host cells. This means that the cell's genome had to predate viruses. Viruses are incapable of surviving without a host cell. So the created heterozygosity and design diversity hypothesis that we hold to as young earth creationists suggests that Adam and Eve and the created kinds were front-loaded with pre-existing genetic diversity and pre-existing functional DNA elements such as ERVs, ALU sequences, and these other classes of retrotransposons. That's why we predict they should be functional. A major prediction of the created heterozygosity hypothesis, okay, is DNA function. Genome-wide functionality. Function in pseudogenes, function in ERVs, function in all aspects of the non-coding regions. And it just turns out that the non-coding regions of our genome regulate all aspects of the gene expression pathway, more fulfilled predictions. So this makes sense. These viral-like elements are front-loaded at creation. And guess what? This had to have been the case in any scenario anyways, since viruses are dependent upon the host cell. So Jackson, why is it so far-fetched to say that viruses and viral-like elements had to start in the host rather than outside? Go ahead, engage that for a minute.
The evolution of viruses is kind of blurry, particularly to me. But viroids, like these really tiny genome viruses that infect plants, were hypothesized that maybe they emerged earlier than life and were kind of attached to somehow attach themselves to life. It's hard to explain,
Jackson,
but kind of the RNA.
Hey, real quick, keep going. Keep going. I don't want to interrupt you, but I've kind of had a sore throat today, so I've been drinking a lot of water, which means I got to run to the washroom, but I can still hear you. So This is your chance to, go on for a minute. Be back.
Right. Well, I probably don't have a minute, but yeah, that's pretty much all I was going to say. Kind of lost my train of thought. But yeah, what's going on, everybody in the chat? It's pretty late, isn't it? Sorry, Donny, I lost my train of thought. But yeah, the evolution of viruses is pretty blurry. There are no virus fossils except in ERVs, kind of. Jackson, execute order 66. What does that mean? Oh, it's some Star Wars thing. It's 1:30 in the morning. I'm having trouble keeping track of my thoughts right now. I'm just going to wait for Donny to get back at this point. I was wanting to talk about ecologic zonation. That's what I thought we were going to be talking about mostly. Got a bunch of visual aids. Got a trilobite fossil right here. I don't know what kind it is, but it is one. 20,000 species of trilobites. Kind of unrelated to anything we were talking about, but there you go. All right, you're back.
See, it's funny. If people say, oh, Standing for Truth, he just dominates the as in, he takes more time than his opponents. Yeah, I give you guys plenty of time. It's not my fault if you don't use it, right? No, but
Yeah, I'm tired.
That's good. I appreciate that. So, since we've been going nearly 40 minutes on just ERVs, let's wrap it up here. Okay, I want to say that I find I'm not saying from you, just in general, because I've debated biologists on this as well. And I just find in general, the responses from the evolutionists, the evolutionary community on ERVs and on the function associated with ERVs, I find the explanations rather weak. Okay, so notice this. Researchers have recently identified an important function for a large proportion of the human genome that has been labeled as ERVs. They act as promoters, starting transcription at alternative starting points, which enables different RNA transcripts to be formed from the same DNA sequence. Notice this. We report the existence of 51,197 ERV-derived promoter sequences that initiate transcription within the human genome, including 1,743 cases where transcription is initiated from ERV sequences that are located in gene proximal, and you can continue this. Okay, so we're not just talking about a small scale phenomenon. This is, when I first debated Gutsick Gibbon and conspiracy cats, it's always there. Well, it's just a few. So we can assume that those few, those functions were co-opted. It's just uninformative gloss. It's storytelling. This isn't a small scale phenomenon. Notice this: the ERVs aid transcription in over 1/5 of the human genome. These data illustrate the potential of retroviral sequences to regulate human transcription on a large scale, consistent with a substantial effect of ERVs on the function and evolution of the human genome. And I mean, we can go on and on and on about.
Sure, I'm not saying there aren't a lot of ERVs that do something.
Well,
I'm sure there are.
And here's the problem. And you know what? This slide sums it up. The evidence shows these ERVs are functional, but not only that, ERVs have crucial functions that help regulate genes and even determine cell types. I say that all the time. I'm not making it up. It's here. Yeah. Claiming they were co-opted is mere uninformative gloss. To understand the nature of their function, one must turn to the research itself. So listen, you evolutionists, you can keep saying that these functions were co-opted, but we can't observe it today, and there's no technical paper that you can present me because it happened long ago and far away. I mean, I'm sorry, that's just not science. It's not convincing. And I'm still baffled. Yeah, go ahead.
The best thing I can do is kind of defer to, what am I trying to say? De novo mutations, which I know you don't believe in, but there's a lot of, there's a good paper on stickleback fish on genes basically arising from non-coding material and how they know different, that they're younger than these over here, and they didn't actually see them, but they can infer. I know more about that than the age of ERVs and how they know the different ages. But I think a lot of it relies on the molecular clock, which I know you don't put any credence in. And yeah, that's all for now.
No, that's good. I appreciate that. Let me say this when it comes to the hierarchical patterns, though. The fact that we would share more overall with, like, let's say the chimpanzee than, a gorilla or a mouse. Okay, so here's some nested hierarchical, some phylogenetic diagrams you can find online. Shared derived features, right? Shared derived synapomorphies, backbone, lungs, claws, fur, mammary glands, opposable thumb, up to human. We could even narrow in our focus on just the primates, right? Apes and humans. We would share more with the old world monkeys than we would with, let's say, the lemur. This is why I always say, just standing back and looking at the morphology and anatomy of, let's say, a human and a chimp, one is going to conclude that there will be more shared in terms of similarity between the human and the chimp than between the human and, let's say, the salamander, or the human and the lizard. And same thing in the, let's say, the ape family, right? Evolutionists would just say humans are apes, but they're like the great apes are non-human apes, right? So they think it's a surprise that we share more with the chimpanzee than we would with, let's say, the New World monkeys or lemurs. No, just standing back and looking at the morphology and the anatomy, it's exactly what we're going to expect. But that's not even to discuss what we discussed before, that even though overall we share the most with the chimpanzee, there's still some massive differences that the evolutionists can't explain, like the taxonomically restricted ERVs, like the Y chromosome dissimilarity. The human and chimpanzee Y chromosome is incredibly different. As a matter of fact, the human and gorilla Y chromosome is more similar than the human and the chimpanzee Y chromosome. So there's, an inconsistency in the hierarchy. So I'm going to apply this to the ERVs now. So of course, we are going to share more in terms of retrotransposons, let's say, between the human and the chimp than we would between the human and the mouse. Yeah, that makes sense. That's what we would predict based on the design hypothesis. Go ahead. What are your thoughts on that?
What about the Y chromosome? It's because of the breeding habits of the different great ape species. We've talked about that before. I don't think we're ever going to agree on that point. But yeah, anytime anyone says salamander, I get a little bit of post-traumatic stress disorder.
Right. You know what? I should have left salamander out of there, Jackson. I apologize. Okay, let me ask this. I'm kind of curious, and we were kind of debating this earlier, since we got the phylogeny here of apes, humans, old world monkeys, so on and so forth. Okay, last common ancestor of all primates, right? Mm-hmm. And then it shows the various splits. Okay, so let me ask you this out of curiosity. No, actually, I think it was in this slideshow. So we talked about ERVs, and how do you explain certain traits and features that humans have, but for the most part, no other primate does, especially the great apes? Baculum is one of them. Okay, the baculum is one of several heterotropic skeletal elements. Example, bones disassociated from the rest of the body skeleton. It is found in all insectivores, shrews, hedgehogs, bats, rodents, and carnivores, and in all primates except humans. Such wide distribution suggests that it appeared early in mammalian evolution. And let me just go pull it up here, right here. The baculum, the penis bone, is a bone found in the penis of many placental mammals. It is absent in humans, but present in all other primates, such as gorilla and chimpanzee. Okay, explain that from your model.
Well, you're saying that we lost it. I mean, there's no other way to explain it.
Or
on the line.
Or separate ancestry.
Or
I don't understand why you say there's no other way.
In my wiener.
But in all seriousness, yeah. Okay, this is scientific evidence. This is a fact. This is an observation. And people like Aaron Ra or Gutsick Gibbon, they're always like, what separates, a human? We're exactly like them in every way. We've put out funny videos. Well, here's just one trait. So you evolutionists always say, well, you just said it. It's the circular thinking of the evolutionists. Well, evolution's true. So we have to have an explanation, and we just must have lost it. Well, okay. Tell me when we lost it then, because chimpanzees and gorillas have it. Humans don't. So when did we lose it? At what point in time?
I'm not sure I've ever found one with Australopithecines or any of the Homo genus. That may be too small to fossilize in those species. I don't know. But I would think before then even.
What happens if we found it in the Australopithecines? What would that tell us?
It'd tell us that it was lost much later.
I mean, don't you see sometimes the humor in, because we'll oftentimes point out that time is the god for the evolutionist. Time did it. Time did it. Time did it. I mean, it is kind of funny. Like, even when it comes to the baculum, time did it. I mean, that's the god of the gaps for you guys. Time is the hero of the story. What are your thoughts? Is time the hero of the story for the evolutionists?
Kind of. I mean, yeah. But, I mean, it is a necessary part of it. But, I mean, even proving the Earth was a few 100 million years younger would still not disprove evolution. But, I mean, if you proved it was 6,000 years old, it probably would. But, or there was some kind of hyper weird evolution going on.
Okay, so let's say, so apparently but again, just like the ERVs, it fits beautifully with the design model. The baculum, the fact that here's just one difference in anatomy between humans and the gorillas and the chimpanzees, fits nicely in the design hypothesis. We don't have to say that it was lost; it was just never there.
Yeah, but do you know how small this bone is in the great apes?
Fairly small.
It's very small. It's like the gorilla. I don't want to be crude, but it's 2 inches long. So the bone is even shorter than that.
Okay, so you're saying because of its size, it's easier to lose?
Well, not necessarily, but I'm just saying it's a very small feature.
No, I understand
and
that.
It's potentially can be lost.
Okay, what about this one? What about this one? How the hyoid bone changed history. The hyoid bone, which is the only bone in the body not connected to any other, is the foundation of speech and is found only in humans and Neanderthals. Is that true?
I think so. I'm not 100% sure. Probably found in some other of the Homo genus, I would think.
So how come it doesn't exist in, let's say, the chimpanzees, supposedly our closest cousin, and let's say the gorillas, bonobos, orangutans? What happened to it?
Well, they never evolved it. They they went a separate route. They didn't develop speech. They don't have the brain capacity for speech, for complex speech anyway.
Now,
they do have brain capacity for well, it's not necessarily true. They can learn several hundred like lexigram keys and essentially talk that way. But anyway.
So are you saying sometime after the human chimpanzee split, the humans evolved this novel feature separately from the chimpanzees and gorillas?
Yes.
When? How? Evidence?
Good question. But I don't know much about the hyoid bone, so, or human speech, or the evolution of human speech. That's a given type question.
Okay, so again, well, and I've heard their explanation, just like with the baculum. It's either it's a gain after the split, or it's a loss, before the split, or a loss after the, a loss or gain, but it's always, too far back in time, to be able to provide us empirical evidence. But just like the ERVs, and the fact that they're incredibly functional in so many different ways. Okay. The hyoid bone is incredibly functional and necessary in terms of speech. Just like the FOXP2 gene is essentially the same in humans and Neanderthals, but yet vastly different. Actually, no, it's only very slightly different in the chimpanzee, but yet the function of it, which helps in language and speech capabilities, obviously is massive when it comes to humans. And yet I think it only differs by like 1 or 2 nucleotide sequences or something. Don't quote me on that one, but I do have some slides on that from the chimpanzee. And yet that minor difference results in what? This massive change when it comes to speech. Okay, so that one was gained. The baculum was lost. Okay, what about this one? Humans have different skin colors because we have different amounts and kinds of pigment of the pigment melanin in our skin. Our closest living relatives, the chimpanzees, have pale skin without melanin underneath their dark fur. How did humans get melanin? And chimpanzees, how come they're without it? What
Well, I
about
think
that
that
difference?
kind of answers the question. Underneath their dark fur, I mean, they don't really need melanin with fur.
Did we split from a common ancestor with the chimpanzee?
Yeah,
What happened to our dark fur?
that would have gone away, and there would have been some kind of coping mechanism for the UV light after that point.
And then the melanin was gained
Yes, I would
in the human
imagine,
line.
yeah, that would be my explanation for it.
Have we observed that type of novelty today? The evolution of melanin in skin, let's say?
Well, you can observe like the loss of pigment in like cave fish, sort of similar in a way.
Okay, so the loss of pigment, the loss of pigment doesn't explain the origin of.
No, but
Okay, what about this one? In total, we have spinal column size and shape, curvature, different number of ribs, cranium size differences, chin in humans, not in any primate, foot anatomy, pelvis angle, lack of. I think this is a huge one. Lack of primate baculum and different hair on bodies. There's one. Okay. What about the vertebra? How do you explain? Yeah.
Well,
how do
the Australopithecines kind of bridge a lot of those gaps.
The Australopithecines bridge a lot of those gaps. How so?
Yeah, they do. What? Like like the shape of the spine. The shape of a chimp spine is an arch basically, and human and Australopithecines are kind of a slight S. And just down the whole list, really, it's like that. Basically, from the neck down, Australopithecines are very human-like.
And that's where we actually get into a whole separate discussion on the nature of the Australopithecines, especially with afarensis, because I just rewatched. It's not a lecture; it's a series that famous paleoanthropologist Lieberger has done, and he did one on species, the definition of species, and just how difficult it is, especially when it comes to just mere morphology and anatomy. And he talked about how Australopithecus afarensis is probably a chimera species. Word for word, I've actually got him quoted in a slide here from the video, because a lot of people haven't believed me. And he admitted that afarensis is a mixture of Australopithecus and human bones from the Homo.
Does he mean human bones, or does he mean other Australopithecine species?
Homo.
Homo habilis, maybe.
He could mean habilis, erectus. Either way, it's still chimeric, as in it's an artificial species. It's a mix of different bones from different species. And he said Homo and Australopithecus, but Donald Johanson admitted too that the Homo genus existed as far back as afarensis. And the new article that came out recently that I was showing you, where there's coexistence between Paranthropus, Homo, and the Australopithecines, and the Homo being erectus. What are your thoughts?
Well, the early erectus, ergaster, that version of it. Some call it ergaster, some just call it erectus, but it had a much smaller brain case and more primitive features than later erectus. But habilis and Paranthropus did live alongside afarensis toward the end there, but they never lived alongside something like Anamensis or Africanus, something like that.
Okay, so would you then, based on what we're talking about here when it comes to the Australopithecines and the extensive range of differences seen in the bones, which obviously would reflect anatomy and morphology. Would you look to the sexual dimorphism hypothesis then to explain that?
Differences in bones.
Yeah.
Well, there's the Lucy specimen is 3 feet tall,
Right.
and what's attributed to a male is 5 feet tall. So that's possible.
Well, I know that they've invoked sexual dimorphism for the,
especially Paranthropus, yeah, because of
especially
the
with the more robust Australopithecines. Well, they'll say that the larger human-looking bones, they would associate that with those with larger male Australopithecines, and the smaller ape-looking bones, they would say that those belong to females that specifically lived in the trees.
Well, with Paranthropus, they're really talking about the skulls that are vastly different between males and females, I think.
But this is what's interested in is, and I've got a ton of quotes and a ton of articles and papers here where you see these paleoanthropologists pretty much contesting each other's finds, where you have arboreals, okay, those paleoanthropologists that would suggest like Lucy and a lot of these Australopithecines are more so arboreal creatures, tree dwelling. They would emphasize specific bones that look very apish, right? While others in the anthropological community would emphasize some of the more isolated bones that have been found that look more human, right? Say, look at this isolated bone. You know, this is more human-like. They associate it with, you know, an Australopithecine. And then they'll say, see, this is evidence that, you know, this was a bipedal creature rather than a creature that spent most of its time in the trees. But then you have other paleoanthropologists, Lieberger being one of them, who suggested at least Afarensis is a chimera species, which means we're looking at isolated bones that may not even belong to the specific Australopithecus. It is the point. And here's the last thing I'll say. The sexual dimorphism that is seen and invoked, from my understanding, is a type of sexual dimorphism that has never been seen today. What are your thoughts on that?
Maybe in humans, but like gorillas have a type of sexual dimorphism in
They
the skull,
do.
like Paranthropus have or had. But there was one specimen, and I've brought this up before, found in the Sterkfontein cave of Prometheus or Africanus, one of the two.
Australopithecus prometheus?
Yeah. That was found pretty much complete and articulated. So, and its arms do have features of tree dwelling, but everything else about it says it walked upright. But and from this fossil, actually determined its legs were longer than the arms, which would have meant that knuckle walking would have been pretty much anatomically impossible for it to do for any amount of time.
How many of the foot bones did they find in Prometheus?
Not very many. They found the ankle bone and a few other bones.
How many bones exist in the feet?
I'm not sure.
A lot.
Yeah.
So if they're only finding a couple, then obviously there's going to be a lot of inference made in the reconstruction of it, which the portions of Prometheus. I'm sorry, go ahead.
Well, it's not even about the foot. It's everything else. Just says, even without the feet, this thing walked upright.
One specimen of Littlefoot was found, right?
About
95% complete, but one specimen.
The guy who discovered it considers it Prometheus.
Others Africanus.
It may just be an early version of Africanus, basically.
Right. From what I've read, some say it's Africanus, others say it's its own Prometheus. Because, and this is what I like to point out, with the fossil. What's that?
There's some differences in the skulls.
Right. Well, and with the fossil record, when you're just looking at morphology and anatomy today, and I oftentimes point this out, that there oftentimes exists more variation within the same species than across species. So if you're just looking at a fossil or a bone, in light of this, it's kind of erroneous with what the splitters do, lumpers and splitters, okay? Where even a lot of paleoanthropologists that are lumpers would admit that, you know, the variations seen in Neanderthalensis, Erectus, Heidelbergensis are really just kind of the same species, but just variation within that same species. They lump those variations in together, while, you know, your splitters, your paleoanthropologists that would fit the more splitter category, they would take any minor variation, like with Prometheus here, like the finder, he wants its own name, he wants its own species. So, you know, this minor variation, we're going to give it a whole new name, when in fact a lot of what we see in terms of the Australopithecines, I mean, it's the same type of creature, the same type of ape creature that, sure, I'll give you is different than the apes today, right? Different than the apes today because they're extinct, but variation within the same creature. But they're just naming it different species based on what I would say is minor morphological differences. What are your thoughts on that?
Well, you may be right to an extent, but there were at least a few Australopithecus species. I mean, Anamensis, Africanus, and they're not really going anywhere.
So I've done, in terms of the separate ancestry model, I think it's pretty easy for us to identify, you know, what is human, what's an ape, what's even an Australopithecine, and demonstrate separate ancestry. Now, as I always point out, with bones, with fossils, we're only looking at morphology, anatomy. We don't have the genetics, which means there's going to be a lot of interpretation, inference. In the fossil record, we know that it's very fragmented. There's huge gaps, and usually the bones are found disarticulated, and a lot of the important parts are missing, or even just like crushed, where there involves a lot of reconstruction. So that's not foolproof, but the genetics is the best way to answer the question of ancestry and determine what is and what isn't related. And looking at a mix of that, and I can make predictions. The human and Homo group, Neanderthalensis, obviously, Homo sapiens, Denisova, Floresiensis, the hobbits, Erectus, Naledi, Luzonensis, Heidelbergensis, okay? All human. And I would predict the ones that we don't have the genetics for. Okay, here's a novel testable prediction from a young Earth creation perspective. If we do manage to get the genetics, some DNA from, let's say, Floresiensis, Erectus, and Naledi, I would predict that it's going to reflect what we see in Neanderthalensis, as in we'll find evidence for inbreeding, we'll find massive rungs of homozygosity, because I believe that these specific people groups like Erectus, Naledi, and Floresiensis and Luzonensis are human people groups that experienced rapid genetic degeneration and morphological degeneration due to reductive evolution. So we can see that in Neanderthals because we have their genetics. We know they were highly, highly inbred. We know they accumulated a lot of deleterious mutations. We know that they have these massive rungs of homozygosity. We know that. But we don't have the genetics of Floresiensis, Erectus, Naledi, and Luzonensis. But I do predict that if we get the genetics, that's what we'll find. That's a novel prediction. Ape-like Australopithecines, Africanus, Afarensis lucy, and Ramidus. Ardipithecus ramidus, but artificial species, wastebasket taxon, Australopithecus Sediba or Sediba. I've heard it pronounced different ways. Homo habilis, Australopithecus afarensis. So I put afarensis here because although I would say afarensis, you know, can be its own species, since there are some bones, including a baboon bone that was missed for, I think, 40 years. They finally realized, oops, this bone doesn't belong. So I'd consider that part wastebasket as well. So when it comes to phylogeny, when it comes to ancestry, I believe I can and have shown a line. I said a lot there. Go ahead. If you want to critique that, give your thoughts, Jackson.
Well, I think when you're comparing something like a chimpanzee and then something like a Neanderthal, and you find something like an Australopithecus, you're going to have to put it in the middle. It has traits of kind of both lineages, so to speak.
Not necessarily, because your Australopithecines are going to be more, and I've got a slide here, they're going to be more ape-like. Okay, like you said, Lucy was what, 3.5 feet tall.
Yeah, but the
they have
male is 5.
Locking wrists. They're I think it's their ear canals as well. I've got a slide here that, where is it? What are your thoughts on this, Jackson? Hold up here. Right here. Semicircular canals better suited for walking on all fours in. See, we can look at the ear canal and determine, like, for example, when it comes to balance, like, you know how you can get like Meniere's disease, or you're off balance if you have an irregulation in your ear canals. So we can actually determine how a creature walked and balanced themselves by examining that. And when it comes to Afarensis had semicircular canals better suited for walking on all fours. What are your thoughts on that?
Yeah, I've heard an argument for this, but I can't remember what it is. I know that's a total cop-out, but I don't really have anything to say about it.
No, it's not a cop. This is just an informal discussion. It's fine. It's not like it's not formal. Right here, vertebra belonged to an extinct baboon. Yeah. Reconstruction of for 40 years though. 40 years, this belonged in the Lucy specimen. And then eventually they realized, oops, that belongs to an extinct baboon. The fingers of Lucy's kind were curved for tree life. Although curved fingers, I don't think is the greatest piece of anatomy to look at, because we do know that disease, pathology, degeneration can result in curved fingers as well.
Well, their arms were more chimp-like, but their legs, their hips, their spine were all very on the human spectrum.
But remember, when you guys say we've got 400 specimens of Afarensis, you realize this is what you mean by 400 specimens, right?
I do. Yeah, there are a few partially complete skeletons, and then it's mostly just fragments.
I mean, when I was in college and when I was an evolutionist, I was fascinated with this topic and still am. That's why I focus a lot on human evolution. When I heard 400 specimens in college in biology, I assumed, and I know a lot of students assume, wow, they have like 400 full I mean, you think of like the full creature and all its bones, 400 of those. You don't really think like 400 isolated bones being your specimens. Isn't that kind of like a little deceitful, misleading, do you think?
Well, I don't know if they're doing that on purpose, but I mean, to say they're 400 full skeletons, that would be deceitful, yeah.
How many full specimens do we have of Neanderthal ancestors?
Neanderthals, I'm not sure. There are a good number of those.
Yeah, a good number of them, where we got the genetics of them. I mean, Neanderthal ancestors, we can make some solid conclusions, but I'm telling you, when it comes to a lot of these Australopithecines, Lucy, even Lieberger is saying we're looking at a chimera species. And this is what I wanted to ask you specifically and get your thoughts on this. People are going to be interested in this one because this is raised as one of the best example of a transitional form. First, I should ask, and I'm not trying to put you on the spot here. I'm just genuinely curious. Have you read this paper yet? Have you read the paper by Elevin and the lumbar spine of Australopithecus Sediba? Have you read this paper?
I've seen part of it. I think you brought it up before.
So how do you address, okay, and Ella Bin and Yoel Rak, I mean, these are experts in their respective field. And they say the phylogenetic scenario that Berger and Berger al. propose, in which Australopithecus Sediba is a link to our evolutionary chain, ought to be ruled out. This isn't from Young Earth Creationists. because one of the Malapa mandibles is too derived to be positioned in the human lineage. And they go into, like, for example, here, the lumbar spine of Australopithecus Sediba indicates 2 hominid taxa. Look at this. The coexistence of Homo and Australopithecus in early South African sites is not, so it's not even rare. Okay. It's not like, well, it's rare. No, it's not unusual. Not unusual. The coexistence of Homo and Australopithecus. And people always say, well, what do you mean a chimera species, artificial species? I mean, like, you know, what happens is like an ape and a human are running at each other, and then bam, they just blow up and all their bones get mixed together. You know, this rare. No, I mean, they're pointing out it's not unusual, this coexistence. And even they point out that is claimed to exhibit many features that Lee Berger, that suggests that it represents an intermediate taxon between Australopithecus africanus and Homo. However, this isn't young Earth creationists. A careful assessment of the fossil remains attributed to Australopithecus sediba lead us to conclude that, as in other South African caves, 2 species, so again, another artificial species, are present. One represents Australopithecus and the other Homo, which I would say is probably more like a Homo erectus. But yeah, what are your thoughts on that? Do you think Sediba is a good line of evidence for ape-to-man evolution?
Well,
yeah, go ahead.
Even if it is a chimeric species, the bones are still unique enough to be considered its own species. So it's still Sediba.
No,
in that regard. Even if they're somehow they manage to be chimeric.
No, because you're looking at
part of it.
Okay, notice this from the paper itself. And I actually just reread it the other day when I was making these slides, so I highly recommend it. In fact, their Australopithecus sediba represents a mixture of 2 hominin taxa, hence transitional. So even Ella Bin and Yoel Rack, okay, two experts in the field who've actually like analyzed Sediba more than anybody. I mean, most people in the field, like, it's not like they're analyzing Sediba in the same way that Ella Bin and Yoel Rack here were in terms of their research and then their subsequent findings. They're admitting that the only reason why it can be claimed to be transitional, I mean, this is their words, not mine, is, and they even point out, to ascertain the origin of Homo, we must delve more deeply in the geological layers. So they're saying, no, it's only transitional artificially because it's a mixture of 2 hominin taxa, and humans would fit in the hominin category. So yeah, you have, from my position, if we're looking at an erectus bone, it is going to be anomalous because erectus is a degenerated form of human that experienced reductive evolution. So yeah, you'd be looking at a bone that you would say is Homo, and you would say, see, it's mosaic or it's basal or it's primitive, when in fact it's pathology, it's reductive evolution. So where you say basal or primitive, we would say degenerate, pathological, or diseased. I said a lot there. What are your thoughts?
I mean, even if Sediba turned out to be a total waste-specific taxon, as you said, it's not really even on. It's like an offshoot, considered to be an offshoot of the human lineage. And I would just go back to Littlefoot. I mean, there's just no arguing that that's a chimeric species.
Well, we'll go back to Littlefoot in a sec. I think Littlefoot is, I think there's not enough evidence to go by. I think the fact that most of the foot bones are missing, and we got a lot of foot bones, and you admitted there's like 4 found, and a lot of which you see in the spine and the other bones. We can't really conclusively say that this is something that's mosaic, something that's transitional or bipedal, but we can examine that more in a little bit again if you want to go back to that. But sure. I do want to point out the problem is Sediba is raised as, or elevated by most evolutionists. Look at Gutsa Gibbon. I mean, she's probably the #1 go-to when it comes to this topic, and she's got a video titled "The Best Transitional Form." But yet, no, okay, the evidence. I mean, here's an article based on the paper. I'd highly recommend just go right to the source, read the paper. Okay. But they point out human missing link fossils may be jumble of species. So it's not like if it turns out to be. No, it is. I mean, I haven't seen a rebuttal. I haven't seen like a better, I haven't seen any better research on this that's contradicting the findings of Yoel Rak and Ella Ben. Have you found any counter to this from anyone in the paleo anthropological community?
I've been studying unrelated stuff here lately. I've been doing a lot of debates.
Yeah, you've been doing a lot of different. You've been doing a lot of different. I do want to point this out that you pointed out earlier, and you're right. New findings as early as look at 2019. Australopithecus Sediba, not likely human's ancestor.
Right.
Temporal evidence shows Australopithecus Sediba is unlikely to be the ancestor of Homo. New stuff, and this is why I disagree with a lot of the baraminologists in the young Earth creationist community that have done baraminological. That's a hard one. Say that 10 times fast. Baraminological assessments, analyses on various traits and characteristics of the bones, supposedly that belong to Sediba. And there's been a few young Earth creationists that said, well, based on our assessment, it's human. When, I mean, there's a lot of ape-like, Australopithecine-like traits and characteristics associated with Sediba. Wouldn't you agree? I mean, if it's real and not an artificial, I think the data is conclusive that it's an artificial species, but some in the young Earth some in the young Earth creationist community. Yeah, go ahead. What are your thoughts?
The arms, pretty much all. I think all of the Australopithecines, the arms are very ape-like. I mean, of course, going to be ape-like.
The hands are nearly completely human-like in Sediba, and that's a problem too. With Sediba is there exists a lot of anatomical contradictions. It's a very contradictory. It's not like a smooth mosaicism going on. There's just a lot of contradictions in the anatomy and the bones associated with it. But I would just side. I've read the paper in full. I've studied it intently from Yoel Rak, Ella Ben. I think it's strong. I've heard some evolutionists say that one of the authors went back on it. I looked hard and wide, far and wide for that, and I couldn't find anything like that.
I might email him and see. That might be interesting to do.
Yeah, I'd be curious, and I don't think that's the case. Let's say worst case scenario, it was the case. Maybe a lot of peer pressure I mean, if you go read I don't think so, because it's a very thorough study. And I just look, man, we've been going an hour and a half on. We've touched on a lot. Yeah. So far we've actually touched on everything we wanted to, except for I think ecological zonation. So if you want, we can get into that a little bit.
But, totally unrelated subject, but yeah, you're talking about the fossil record tomorrow, so I kind of wanted to talk about that.
All right. Well, let's let me see, what time is it? 3:20. So I'm wondering if anybody would even want to join. I'll put it
join in,
because I did promise that we would do a little bit of an open mic on this too. I put the link in the chat temporarily. Again, like I said, throat probably all we had a crazy week last week with shows, so maybe all the talking, I'm not sure, but I've been drinking a lot of water. So again, 30-second break, gentlemen. We're going on an hour and a half. Jackson, we pretty much just had a full, I mean, that was hour and a half flies by when you're talking about the hominid fossil record, when you're talking about ERVs, so you're a champ. Okay, 30-second intermission, guys, and we will be back, we promise you. Jackson, we're back.
All right.
We're back. So, are you pretty excited about your upcoming debates or what?
Yeah, I am. Especially the fossil record debate.
Yeah. Fossil record debate with
T-Rock.
Then you've got round 5, chapter 5 debate with Kent.
That'll be fun.
I think we're also working at getting you a debate with Matt Powell. Let me see. Yeah. I saw a comment, Jackson, real quick in the chat. I wanted to. How many debates have you had so far, Jackson?
I don't know, somewhere like 10, maybe more.
Yeah, you're getting up there. You're getting pretty soon. You're going to have a new record when it comes to evolutionists, the evolutionist side. You ask the good questions. That's why you're fun to see.
Yeah, not sure I'm the best representative, but I'm doing it.
There's nothing really all that fun talking about like human evolution, the hominin fossil record, and paleoanthropology with an evolutionist who doesn't really know anything about it. Mm-hmm. So that's why I do enjoy discussing these things with you.
Well, thank you.
Because you do have a lot of knowledge on it, and it's just such an interesting topic. So I saw a comment. I can't remember. It was from Rohini here, who said that Sediba was probably an offshoot, a side branch, which at best, if it's not an artificial species, yes, it is a side branch. We as creationists, we'd have to address why it has mosaic features. Okay. If it's real, I would say it's your best example of ape-to-man evolution. Although I would think, let's just for sake of argument say Sediba was real. Let's even say habilis is not a wastebasket taxon. Let's just say you had those two, and then let's say Prometheus. Mm-hmm. I mean, ape-to-man evolution, the amount of like evolutionary experiments that would be required to take an ape-like creature. Like I was watching a documentary. I've been writing a book as well on this topic. So I've been watching a lot of secular lectures and documentaries. And the documentary that I was watching last night, I think I actually sent it to you on Facebook.
Oh yeah.
They were saying that if you go back even further than the so-called hypothetical common ancestor between humans and chimps. So back 13 to maybe 15 million years, mm-hmm. Right? The ape-like creatures before that common ancestor, but before we split, would have looked something like the apes today. Almost identical, the documentary said. I mean, do you agree with that?
I would say it would look similar.
So it's just kind of, so it's like, so then we could, in a way, imagine, I guess, SpongeBob style, an ape today then, given, let's say 10 million years, could it ever evolve into something similar to humans today?
I mean, maybe it would look kind of human, but it's not going to evolve into a human.
Well, yeah, because the law of monophyly. I know it would be a side branch. You'd have humans here, and then whatever humans branch off to at some point, millions of years down the road, and then you'd have, let's say, some offshoot of chimpanzees. But you're saying at some point it could look very similar to what we look like today.
If there's suddenly a pressure to start standing up more and somehow lose their hair, then they might look a lot similar.
So shouldn't you guys then have thousands and thousands and thousands of Sedibas and habilis type creatures? Like, I just don't understand. Why is it always just a handful that we can sit here and argue? I mean, and I got articles and papers suggesting that the origin of Homo is kind of a mystery. I mean, there's pretty big gaps from where you have like your Australopithecines and then Homo habilis. Everything in between for millions of years, there's kind of like not much. It's kind of fragmented. There's gaps. I mean, and it's bushy. So they'll talk about how it's bushy. There's so many side branches. Shouldn't there be thousands and thousands of examples where we're just like, okay, I mean, we tap out. How can you deny this? What are your thoughts on that, Jackson?
Well, the way I understand it, they were living in kind of a transitional environment where fossilization wouldn't necessarily be that easy. So you wouldn't expect to find piles and piles of them like you would find trilobites or something. But As for the second part of your question, which I'm trying to remember.
Well, shouldn't we? I mean, to go from prior to the split.
But even one, wouldn't even one just do it if it was a really good one?
No, because you would need countless intermediates to go from, as you pointed out, prior to the split, you'd have an ape-like creature similar to the ape-like creatures we see today.
Sure, if you have a snapshot in the middle and it's a good example, why would that not be enough?
So you're saying just one example. Let's say Sediba was real and all we had was Sediba. That would be enough to just, okay, ape-like creature, you know, and then eventually split. Chimpanzees go this way, humans go this way. Well, you'd have Australopithecines, then eventually Homo habilis, erectus, modern Homo sapiens. That one specimen would be enough for you to kind of just bridge all of that?
Well, if you had one in the middle that had transitional features, it would prove that obviously not just one existed. You know, it was part of a population.
Right. But how do you know that population, like Sediba, like they're saying now, just a side branch, it just went extinct, didn't really go anywhere?
Well, if you found more than one species and you start finding them closer to what you think,
but I thought you said you would just need one.
Well, you do just need one to prove the transition's happening. Even Sediba being not truly on the line, it still has features of both. So that would, even that, if you just had that, we would probably say, well, yeah, that's something's happening here.
So do you think Sediba being a side branch, okay, so obviously you have an individual that exists in a population. So that individual that exists in a population, okay, Sediba, Australopithecus Sediba, it's an offshoot. I mean, just like those articles said, right? It's not likely an ancestor Homo. It's been ousted. So it dies off. It's just a side branch, doesn't go anywhere. Where's the branch that actually led to humans that it went somewhere then?
I think it's probably around Afarensis. And the origin of Australopithecines themselves is around Anamensis.
What would you say from Afarensis? It's a question from Sea Science Film Labs, because you know my position. I think Afarensis, although the portions of it that are Australopithecine obviously belong to an Australopithecine. Mm-hmm. You know what I mean? There are creatures called Australopithecines, right? That are an interesting ape-like creature that are different from apes today. But from the Afarensis specimen, what part of it, since I would be on the side of Lee Berger and other paleoanthropologists that would say Afarensis is a chimera species, what part of Afarensis do you believe is transitional or mosaic?
Well, you could take any part from the neck down, really. I mean, you could just say, even if the knee is something else, well, what about the femur? It shows that it was a biped too. What about the hip? What about the spine? What about the ribs?
But how do you know the parts that you're looking at that you think are transitional because they're more human-like are not actually human, since it's a chimeric species?
Well, the odds of them all being human, they're not very good.
Well, and nobody's saying it's a mix, but the select few that because evolutionists would point out, and I actually made a slide because I've been prepping for, but I didn't get it into my slideshow yet. I want to see if I can pull it up here, of the amount of ape-like features. Let me see if I can pull it up here. So I've got a couple debates actually coming up on this specific topic, and so here's a slide that I made here. Afarensis found in a bone bed which had hundreds of bones, including dozens of mammal species, all mixed together. Found a knee joint that was identical to humans, a partial femur, and pelvis that are all human-like, mostly waist down. Found bones, waist up, that are clearly Australopithecine, knuckle locking mechanism, skull clearly ape-like, the size of Lucy, ape-like, 3.5 feet tall, for example. So yeah, so those isolated human-like bones, since afarensis is probably a chimera species, then those few human-like bones are the human bones that are mixed in. What are your thoughts on that?
Well, say the knee, for example. There are 3 good specimens, partial specimens with the knee preserved. So we're saying in all of those 3, the knee showing an upright walker, they're all human.
Well, the knee is not showing an upright walker.
In what way?
Okay, so here's so Australopithecus. So all of these features right here are more ape-like: arms, shoulders, chest, teeth, brain size, hand bones, striding gait. See, at best, you have a creature that has a different gait than apes today, different than humans, but not upright. Because, for example, the line of evidence I pointed out earlier, Jackson, about the inner ear canals and how they're consistent with knuckle walking or not walking on not being bipedal, the toes, teeth, development. Point is, the majority of these specimens related to the Australopithecines are more ape-like, with some interesting variations, and then some actual human bones mixed in from the Homo. And this isn't just Cretaceous making this up. I mean, I've shown you quote after quote from evolutionary paleoanthropologists that admit this.
Sure.
Yeah, I don't think I answered your question. I don't want to look like I'm dodging. So you're saying the knee that's broken up, you're saying there's the different isolated fragments of the knee?
Well, there's a knee in 3 specimens. That's the 3 partial skeletons that are really good for afarensis. That all preserve the knee.
Right here. Of these specimens.
Well, no, there are a couple more that aren't really on the table there that you can't really make out. Maybe there are.
One quote that I find interesting right here from Richard Leakey. I see no reason that bands of Homo would not have killed and eaten robust Australopithecines when they could, just as they killed and ate antelopes and other prey. I mean, humans, especially in desperate situations. Let's say, according to our model, okay, you have after the flood, you kind of have you have culture, you have everything that pretty much has to be you got like a Stone Age in a way, right? You got to start making stone tools, rebuilding civilization, reestablishing culture. But humans, they did not follow God's command to spread out in all parts of the world. Animals did, though. So you have these ape-like creatures, let's say these Australopithecine-like creatures, spreading out. Then eventually humans spread out in all parts of the globe after Babel. You have many subpopulations, unfortunately, becoming isolated. What occurs is inbreeding, founder effects, mutation-related genetic degeneration. These recessive mutations come to the forefront, leads to morphological degeneration. You get these anomalous features that look, basal, when in fact they're just the result of pathology, disease, Naledi, Floresiensis, Erectus, Neanderthals. These are isolated people groups, okay? But other people groups that are trying to reestablish civilization and culture, and humans have always done this throughout history. When they're desperate, they'll eat anything. Apes, monkeys, Australopithecines. So what happens? They eat, the bones get laid there, and eventually the humans die. They get mixed in together. They get mixed in with other prey animals. Yeah. What are isn't this kind of interesting? What's your thought on that?
But it's not talking about anatomically modern humans hunting like Anamensis. It's talking about like early Homo erectus or Homo habilis hunting.
Right. Oh, I know.
Paranthropus.
I know. Okay. But that's not to say that those are the only Homo groups that ever ate Australopithecines.
Why not?
Are you saying a Neanderthal, let's say a population of Neanderthal, if they're not desperate enough for food, they're not going to eat an Australopithecine ape?
Well, they didn't live in the same time period or the same geographic location.
Right, but okay. But that's circular right now. So the time frame would go back to the ape-to-man evolutionary starting point. But my point is, bands of Homo, I would say Erectus is a human. So yeah, when a lot of these paleoanthropologists want to associate Erectus with these early Homos that are mixed in or overlapped with the Australopithecines, yeah, that's a human. I mean, for example, here, where is it? Homo erectus may have been a sailor. Homo erectus may able to speak, purposeful navigation, able to cook. I mean, everything about Homo erectus, the fact that they are a sophisticated people group, shows that they were human. And notice this. Extensive coexistence evidence found at all three. I'm going to show the source for people. This is a lecture from Christopher Roop. Some of his slides, which I thought were fantastic. So like, I got to save these. Extensive coexistence evidence found at all three major East African hominin sites. Thousands of butchered bones, diversity of stone tools, human footprints. You would just say the Laetoli footprints are probably like Anamensis or Africanus. Human bones, windbreaker shelter. Shelters that clearly only, and here's the thing, habilis, if a wastebasket taxon, that means you have Erectus bones and Australopithecus bones. So when people say, well, Homo habilis ate bands of robust Australopithecines, to me, what that's saying is Homo erectus ate robust Australopithecus bones. Does that make sense?
Yeah, I follow.
But I'm sure you probably want to get to another topic. So I know we could talk about this all day. So,
yeah.
But I don't want people to think I'm, I want to give you the last word. I don't want to, people are saying I'm slap and run kind of thing. So give some final thoughts on our first 2 hours here on human evolution, and then we'll move into something else.
Yeah, it's just interesting to talk about. We've kind of had this debate like 5 times, though. This exact pretty much debate. I don't think we're ever going to really agree on whether or not Sediba was a real species or not. But
well, do you think Yoel Rack?
What?
Do you think? I know I said I'd give you the last word, but I'm curious.
Yeah.
So, on record, do you believe that Yoel Rack and Ella Bin are wrong?
I don't know if they're wrong. I don't know if they're wrong in the strictest sense of being wrong, but I think there are a lot more anthropologists who say this is a unique species.
Crazy. He says, you don't slap and run, Donnie. I hope you don't recall my remarks from last time I was just playing. No, I was, because I realized at the end there, I threw out a lot of points and I wanted to make sure that you had time to address it before we moved on. Anybody new in the chat? We've got our new, heterozygous Adam and Eve shirts. So I'm pretty pumped because I just got mine in, and these shirts are going to make my choice now of what to wear during all these events we have planned easy because I'll just end up wearing one of these shirts. Nori Davis says, will Jackson debate me on the resurrection? That's not really one of your specialties,
no,
right?
I mean, I don't care if the resurrection happened or not, so I don't really have a dog in that fight. I mean, I obviously would care if it happened, but I mean, I'm not going to argue that it didn't happen. That's what I'm saying.
So we did go, whoops. I am putting the link in case anybody had any points they wanted to make on this topic. I know we went late tonight. So I just want to, so I'm posting the link again. Whoever joins, I just want to warn you guys, we're keeping it on topic, ancestry and things related. And I think we're about to go into, I see Alec.
What's
Alec
up?
Cox. Good to see you, my brother. My man,
what's up, Jackson?
How we doing?
Hey, hey, all of my ancestors that I know of were upright human primates. It's kind of, as far as I go back, I mean, we have a long lineage all the way to the crown in Scotland. So, yeah, they kept great records. My daughter's actually doing the genealogy thing of ancestors. So I don't remember, coming from any little primate out there in Africa or a gibbon somewhere on the planet. Nothing like that. But hey, we can continue with the Mickey Mouse story. of ancestry, and heterozygosity is such a good thing. Love
Well,
saying
Alex,
it. Don't you love saying it?
I love it.
Just love saying it, man.
Hey, I've been saying it for 3 years now, and RJ Downer has been having nightmares almost nightly for 3 years.
Yeah.
Heterozygosity, isn't it? Just
do you know what's
so nicely?
Do you know what's in the middle of that word?
Heterozygous.
Zygote. There you go.
Zygote. There you go. Zygote.
Anybody who's had children, you know about the zygote.
Yeah, we know about the zygote.
And if you look at it, if I was to ever get a tattoo, that would be my tattoo.
Heterozygosity.
I want to make one tattoo on my arm.
Well, it goes back to
I got a couple.
Yeah, it's a
story.
Before
I was a Christian, though. Before I was a Christian, and realized I shouldn't get them. But I'm stuck with them now. So,
you're forgiven, bro. I mean, just don't get that mural of that Mexican guy on your chest.
I wanted to get a tattoo of the heterozygous Adam and Eve.
There you go. You can
cover up the Charles outline on my arm.
Yeah.
What do you think?
No, you should get a tattoo of time. My God.
Oh, wow. That would
be good.
The God of the evolutionists. Time can solve anything. Time,
I feel like that would make a better
Speculum lost. What's that?
That would make a better T-shirt, I think.
That's you know what? That
That'd
actually
be awesome. That'd be awesome.
I'm sending that to Matt right away.
Yeah.
Matt's wife, our clothing line designer. So if she's listening, Matt, we want a new shirt that says "Time, the God of Evolution." Time can solve. But you know what's funny? Jackson? Time is not the hero of the plot for evolution.
Yeah, genetic entropy.
Right. See, the more time, the more mutation accumulation, the more disease, the more cancer, the more extinction and death. But I know you disagree with that, Jackson, so that's fine.
Yeah, I was actually talking about that with my brother the other day. We were talking about when they're going to change from 14.7 billion to 50 billion because they find some new solar system out there that echoed back something. Because we still have, was it not Voyager, but the other one's still floating around somewhere?
Well, that's why they're constantly pushing back the time for the split, the hypothetical split between humans and chimpanzees. That's why you'll oftentimes hear me and Jackson talk about the split. And Jackson, you'll say things like, well, maybe between 6 and 13 million years. I mean, that's kind of a big gap.
That's not like a weekend, Yeah.
And it's because the evolutionary community are recognizing the fact that fixation rates are slow. And so if you have, this many hundreds of millions of DNA differences, even if you went with the 98 to 99% similarity, which that's been refuted, but let's just say that would probably be about 40 million to 50 million DNA differences. So even there it's not enough. 6 million years wouldn't be enough time to fixate. Fixate just means to get stuck in place, those DNA differences after the split.
Right.
So the evolutionists have to keep pushing it back. Well, okay, maybe 10 million years, maybe 13 million, maybe because they need more time to fixate those differences. Is that right, Jackson?
I think it's more mitochondrial mutation rates they're tracking, not necessarily accounting for overall mutations. Trying to push it back to gain time.
What about the Y? The Y chromosome is nuclear DNA. So the Y, right? I mean, you have the autosomal DNA, and then you have the uniparentally inherited DNA compartments, mitochondrial DNA, which is inherited from our mothers. Mitochondrial DNA is not nuclear DNA. Y chromosome DNA is nuclear DNA, but it's still uniparentally inherited DNA. But there's massive differences in the nuclear DNA, including the Y chromosome and the mitochondrial DNA. So I don't think
mitochondrial DNA is just a part of the DNA structure that's needed for. Yeah, it's not. You can match the mitochondrial DNA to your own DNA because it's just a small snippet of it.
Well, and they need all that time to fixate all of those differences in the nuclear DNA compartments, in the mitochondrial, because the mitochondrial DNA, we had Dr. Rob Carter on here, and he was talking about how on his computer he's got, I think he was saying like thousands of sequences of mitochondrial DNA, and the human mitochondrial DNA is nothing compared to the chimpanzee. There's huge difference. You can't even line them up, he was saying. They're so different.
Well, the other thing is the regular human cell, not talking about skin cells, are different. Your muscle cell can only last a day outside of the body. A lot of people don't know that muscle cell has multiple nuclei, but the it can only last like a day outside of the body. Lung cell tissue can only last a minute outside the body. So everything inside your body exposed to air will die.
Did you know that they won't sell fetal bovine serum to civilians, so I can't make a human cell culture?
Did you get that target?
They won't do it.
Well, you're probably not involved with the university.
No, I'm not.
Yeah, well, that's number one. Not your average Joe can just go in, I want to get some DNA. Unless you're at one of those places in Las Vegas or something, you can get a lot of DNA. But no, you have to be a licensed carried official in order to get that stuff, because you don't want it. It's a lot of these companies make a lot of money selling this stuff to universities that's already prepared, already cooked, already ready to go. And so what they find in a lab is already prepared. Oh, look what we found.
Yeah.
But they can't keep it together. So that's part of the lie of universities. But if you wanted to get some, just get a cow.
Blend up its embryos, I guess.
Yeah, just get a cow, get 2 of them, and work on that. And it's very easy. You can eat it after you kill it. You can eat it after you kill it.
Yeah. Take a long time.
Yeah. No, it's okay. Yeah. I told you the story about my dad raising cats for medical school, right?
Yeah. Yeah. You said that.
Yeah. And so raising a cow is pretty quick.
Yeah.
They only have to be 2 years old to actually, I believe, breed 2.5 or something like that.
Yeah.
But how do you get the green grass into white milk? I have no idea. But yeah, the problem with a lot of the cell structure is a lot of what people put down on paper. We already had that discussion, but it has to be done, and then it takes a long time for that paper to be made, right? So it could take a year, it could take 2, could take, it doesn't take a month to do a lot of these studies. So the next problem is somebody to come in and piece it together and see what the actual paper says, and then do it on their own, right? So you're talking probably 2-3 years down the road, before that's even done. And then you're talking probably another 2 years before the rebuttal paper comes out. So you're talking about 5 years. And that it's on purpose, of course. But, it's not like, one university is doing this, they're doing it at the same time, they come up with 2 opposite things. It's normally not done that way. It's like every institution gets it filtered out. That's why some universities get, their grant money, say, no, we can't do that. We're already funding it for some other university. So they have to come up with some different avenue for them to do a similar thing, but it's not the same thing. Unless you get something like what BioLogos is doing, or, a lot of private funding involved. Once you get private funding involved, it's generally more reliable than university funding. Just to let you know.
Yeah.
Because, private, that's actual real people's money. So there's a lot of, it's not government, tax dollars that's divvied out to universities, set aside for science projects, that money. It's private money. So they demand results.
Yeah.
And, so you can look at petroleum exploration on stuff like that. More monetized stuff, not so much science, because you're talking a lot of money. I mean, that's a lot of money to get involved in all that stuff. You can't have a day job and try and finance your studies with a day job, like say Tesla or somebody back in the day.
Yeah.
Those days are gone. Just like you said, you have to go to a certain place to get certain ingredients, right?
Mm-hmm.
And if you don't have the credentials,
that too.
Oh yeah, I mean you need a lab too.
Yeah.
You have to make sure it's all sterile. I mean we can do a lot of investigation in our kitchen sink, but it's not really sterile.
Yeah.
Room temperature about 65 degrees, all that fun stuff.
Mm-hmm.
So
where are you saying, Don?
Go ahead, Alec. I apologize.
No, I was going to say, what were you going to say?
Well, I wanted to show this. I know you've seen it before, Jackson, but especially for anybody in the audience who hasn't seen it. A lot of what we were talking about when it came to the fossil record and the so-called examples of ape to human evolution when it comes to the hominids or hominins, we were discussing how a lot of the anomalous features can be attributed to pathology and disease and degeneration and reductive evolution, mixture of all, rather than transitional or mosaic. And there's a paper from Eric Trinkos that I highly recommend titled An Abundance of Developmental Anomalies and Abnormalities in the Pleistocene People. And he points out, it is apparent that the Pleistocene human fossil record is characterized by a plethora of developmental abnormalities, some relatively well known, but others extremely rare. The probability of finding this density of rare developmental abnormalities in recent human samples of comparable size is again vanishingly small. So for my model, here's also the genetic cost of Neanderthal introgression, talking about how the Neanderthal DNA is depleted around functional genomic regions and has high levels of homozygosity. So according to an Alec's model too, the creation model, this makes sense because we talk about how pre-Babel dispersal and the post-Babel dispersal, a lot of these people groups that migrated and became isolated, they became inbred, diseased, and they were subject, unfortunately, to rapid genetic degeneration, explaining a lot of their anomalous features. And yet this is what we actually see. Now, of course, Eric Trinkhaus isn't going to agree with the biblical creation model, right? This is the observation. This is what we see. The data is consistent with us. He would have a different interpretation, but he is admitting that the probability of what they're finding, okay, that amount of rare developmental abnormalities is small probability. But for us, it makes perfect sense. That's what we would expect, given how many isolated people groups you would have after Babel. So I don't know, Jackson, give your thoughts on that. And to be fair, I know you did want to talk about the ordering of the fossil record a little bit too. Yeah. So go ahead. I'll hand it over to you. And Alec, you can give your thoughts too, brother, of course.
Yeah, I don't really have any more thoughts on that. We kind of talked that into the ground, that subject. But yeah, the order in the fossil record, I know there are a couple of creationist models for how it happens. Some say hydrologic sorting, some say ecologic zonation. Donny, which do you think it is, or is it a combination?
Okay, just one sec.
Okay.
I thought you were going to talk longer.
No,
I need you to go on a diatribe once in a while. Okay, so I have a slide for this, the different models. So let's see here. So like you said, there are a variety of models. I wouldn't say, I mean, I do hold to the ecological zonation model or explanation, but I wouldn't say that that's the only explanation for the ordering of the fossils. I do believe that we can. So for example, ecological zonation, escapability, a species' ability to escape the incoming flood waters may have played a role in determining whether the flood buried them earlier or later, right? Some species. So I'm not saying, so it's not like, oh, well, I only get to choose this one, but not the other. No, I think all of them can come into play a little bit. when it comes to the ordering of the fossils and the flood itself. But mainly ecological zonation, but obviously some other factors as well, like intelligence and where different forms of life are located at that time on the planet.
So something
yeah,
like
go ahead, take your time.
Something like bottom feeder, bottom dwellers, slow moving animals would be found in like a Cambrian layer, slightly faster, depending on the environment. Also, something like that.
Well, you're probably you're going to have different ecological zones. You're going to have different ecosystems in the pre-flood world. Okay. Different elevations, different ecosystems in the ocean as well. Yeah. And so you'd have an isolate a certain ecosystem, and you'll have things like escapability, intelligence to that ecosystem. You know what I mean? It doesn't really have to apply. Does that make sense what I'm saying?
Yeah, let me give you a really good example though. It's starfish. This is actually my former pet starfish when I was a kid, Bermuda starfish. He kept eating everything, so we just dried it out. But probably shouldn't have done that. But
hold that up again.
Yeah. Bermuda starfish.
Is that your when you look at that starfish, do you think that's my cousin right there?
Maybe. But my point is,
no, I know.
There are 2,000 species of known starfish, and they live from the Arctic to the Antarctic and everywhere in between. They live under the Antarctic ice. They live in the shallows, and they live in the abyss. And obviously, they can't move very fast.
No, they can't.
So
they're going to be done for.
They're in all environments, pretty much. Any conceivable environment. They can't go anywhere very quickly.
The Antarctic is land. It's not ice.
Well, parts of it. There's parts of it that where you can go under. But yeah, it's mostly land. But
well, if you're a flat earther, that's where the world ends.
Right, that's the ice wall.
Well,
so
go ahead, finish your argument, and then we'll
Shouldn't we find starfish in the lowest layers? Or all the layers? Any layer? I mean, we looking at?
Okay, well, so you're saying starfish are found in, well, primarily what layers are they found in, Jackson?
They start in the, I think, mid-Ordovician period.
Okay.
Definitely the Ordovician. I forget when exactly in the Ordovician.
So if what we see a lot of the creatures in the various layers, okay, are not reflective of like large-scale evolutionary history, but more so reflective of communities being buried at different times during the flood, okay, but you're saying the starfish are found in the Ordovician layers period? Okay, well,
to also find them in the Cambrian.
Okay.
I found one on the bottom of our boat one time.
Yeah.
What layer is that? What layer is that? No, I mean, starfish they're not fast creatures.
No.
So yeah, they're pretty, they're hunters, they eat the barnacles and things like that, kind of like octopi. Octopi don't stop growing. Did you know that?
Yeah, they keep growing. They don't live very long though, so
that's why I don't want to come face to face with one of these krakens. But I'll say this. I'll say this, Jackson. What's interesting about the fossil record, and we'll kind of tie this in with your starfish example as well, is we have marine fossils that, and would you agree or disagree? Marine fossils make up most of the fossil record, and they're found in every single layer. Like we find marine fossils, obviously, in the Cambrian layers, but we also find them mixed with dinosaurs. We find them in the highest layers. We find them all throughout the so-called geological record. Agree or disagree?
Mount Whitney.
Yeah.
In California. Still has
marine fossils. Marine fossils found in landlocked areas that are far from the sea, on mountaintops. And the reason why is
plate tectonics, of course. Oh.
Yeah, well, of course the catastrophic plate tectonics, we would have meters per second movements of the plates, where you have the buckling of this rock strata rapidly creating these mountains. So the marine fossils are buried before the mountains are uplifted. Yeah. But what explains that is the fact that we have this global flood, where we have the flood itself founds of the great deep, right? So the flood itself begins in the ocean, in the waters, rips up all of these marine creatures, including these starfish you're talking about, rips them up and buries them on the continents. So these are the kind of creatures that would be first to be buried. And as the floodwaters rise higher and higher, okay, through plate tectonic shifts as well, because you got the continents are moving vertically, they're moving horizontally, okay? And then we have the burial of the land animals closer to the end of the flood. And then that's exactly what we find. But what's interesting is we find the marine fossils all throughout the fossil record, including the starfish that you're talking about, because you pointed out we find them in the Ordovician as well, and today.
But not the Cambrian or before.
Well, okay, starfish not in the Cambrian, right?
Right.
But if these layers and the bodies of creatures that we see in the specific layers are reflective of communities, then that was not a community that starfish were in. I don't know why that would be.
Well, there are a ton of Cambrian fossil sites all over the world. Surely one of them was in the right spot for starfish.
Well, we have very specific groups that are found in the. So, for example, here, let me see, ecological zonation. So, one by one, the floodwaters swept away distinct communities of plants and animals. So what we see in the Cambrian would be a distinct community of plants and animals. And that's actually what we see is distinct. So starfish aren't in the Cambrian. That's just a distinct community that the starfish were not a part of. It transported them and buried them in their final resting place. As the floodwaters' destruction advanced, deep sea communities fell victim first, followed by those in near shore shallow waters, then on the shorelines, and finally those further inland. And then you can yield after this, what we find is these explosions of communities, okay, which I think is fantastic support of the ecological zonation model, because you would say it's reflective of deep time evolutionary history, where we would say, no, these communities were there. Okay, these communities were obviously originally created. They didn't evolve up through evolutionary processes. So the fact that we have the Cambrian explosion, who you just debated David McQueen on, is an excellent example of an explosion of life forms. Before the Cambrian explosion, layers that contain fossils are few and far between. Of the fossils that are found in these layers, in your debate, you pointed to a couple, right? Mm-hmm. Most of them are single-celled algae and microbes, and a few are very unlike any organisms we are familiar with today. The Cambrian explosion represents the sudden appearance of almost every major group or phyla of animal. We also see explosions of life in the post-flood fossil record. These are likely the result of mega, so then you can go into that as well. But the point is, these explosions of communities represent communities that existed and didn't evolve through evolutionary processes during the flood, and then were swept up and either buried in place or thrown onto the continents and buried there, right? Not necessarily buried where they live, because if they're being thrown onto the continents. But I just find it interesting that that's what we see, the burial order of the flood, but we see marine creatures all throughout, no matter where you look. Go ahead. I don't know if you feel like I answered your question, but you can give some pushback if you want. Give me a critique. Go ahead.
Yeah, well, with there's something called the Ordovician diversification. Like I'll give you cephalopods as an example. In the Cambrian, you do have predecessors of later cephalopods like Plectronoceros, and then in the Ordovician, they diversify into all kinds of weird cephalopods whose name I can't think of right now. But it's, yeah, I lost my train of thought. Anyway, if I, the other thing I was going to say, if I put a star,
okay, yeah, go ahead.
If I put a bunch of starfish, a bunch of shark teeth, and a bunch of trilobites in a tub and sweep them all up in succession in a big tub of water, are they going to all form into the same layers with each other and without exception?
Not without exception. No, that's why we do find some anomalies. You can even read in the evolutionary literature when you find out of place fossils, or you'll see a lot of times explanations that involve thrusting, overthrust, to explain why certain fossils got to where they, I mean, like the Ashley phosphate beds, for example, where you have a mixture of all different types of creatures. I think they look to like overthrust that would bring something like a human fossil into the same layer as a dinosaur fossil. So yeah, no, of course there are exceptions, right? It's not like, yeah, there is a general order to the so-called fossil record, but you do find a lot of what the evolutionary community would call anomalies. And we would, and that's kind of what we'd expect too. Yeah, it wouldn't be like no exception. It's a good question.
Yeah.
Go ahead, Jackson.
Well, I mean, like my shark tooth example that I wouldn't shut up about during the Queen debate. I think, you know, they're dense, they're numerous. I think you should find those in the Ordovician, Cambrian.
But here's the thing, though, Jackson, is we have a pre-flood supercontinent. If you hold to that model, if you hold to catastrophic plate tectonics, you have the fountains of the Great Deep breaking open, and then this is where you have plate tectonics. At first, you'd have rapid plate. I mean, 100 years ago, I was watching a lecture today. Nobody believed in plate tectonics until they've gotten enough data to say, okay, yeah, the plates are moving. Okay, they're moving at snail pace movements today, and they can even determine this through satellite imaging on the ocean floor and the crust. Right, they can see cracks that are going in directions indicative that plates are moving and have moved. It was a creationist. I think his name's Antonio Snyder. I have slides on it, but for sake of time, I'm not going to pull him up. He suggested plate tectonics because of the Bible's explanation in Genesis of how the land is in one place, right? Suggesting a supercontinent. So not only do we have different ecosystems, we have different elevations. We have a pre-flood supercontinent for the most part that splits apart. The earth is fractured like an eggshell. Plates are moving horizontally and vertically at the fountains of the Great Deep. And that's actually why we have what's called runaway subduction. We have these plates that are moving through the floodwaters at meters per second movements, right? Meters per second movements through the floodwaters, where you would have two plates coming at each other, and you'd have what's called subduction, where one plate goes under the other one. And that's why we find these massive slabs of cool rock where the crust, some even close to the core, but in the mantle for sure, they have not yet been assimilated to they're still dense, like the crust on the earth, representing that they're cold, they're cool, they haven't warmed up to the surrounding material, which is actually what we'd predict based on the CPT model, where you'd have essentially the recycling of the pre-flood crust that would be cold and the new crust would be hot. But my point is, you'd have, so wherever sharks were or starfish or whatever these pre-flood organisms were at the time of the pre-flood world, we have these different ecosystems, different elevations, right? The Bible says that rivers flow out of Eden. Rivers only flow downhill. So that would suggest different elevations. You'd have humans on one elevation in their own ecosystem with their own plants and animals and fauna. And then you'd have dinosaurs in their own habitat. But once the flood started and continents started moving apart, I mean, anything's game at that point. So are you going to have sharks that are swimming through one ecosystem and shedding their teeth in that moment? How much fossilization was taking place pre-flood? Most of the fossilization would have been during the flood, right, Jackson? I said a lot there. Analyze it a bit. What do you think?
Well, just falling to the bottom and getting covered in sediment would be enough for like teeth.
All during the flood?
Well, no, just during normal conditions. Sometimes,
like during the pre-flood. Now, are you looking at sharks? Are you looking at water ecosystem animals today? Let's say whales, sharks, and the ecosystems they live in today, and assuming that's because remember the flood completely changed the real estate of the earth. The real estate of the earth was completely rearranged.
Right.
So ecosystems today are not the same as ecosystems in the pre-flood world, right? God initially created two people, Adam and Eve, two people, and then populations of animals that filled the globe, billions of animals. So that's why we predict, well, the fossil record should reflect that. Very little humans to none, and billions of animals. And that's what we find, billions and billions of fossils that require rapid burial anyways. But yeah, those ecosystems, it was a whole different world. So, well, my question to Jackson is this, and then Jungle, I'll let you jump in because Jackson and I have been debating for a while. Are you assuming the ecosystems of today, when you look at sharks and starfish and things like that, would have been the same in the pre-flood world?
Well, say you have a supercontinent. Around the edges, you still have the coast, and further out, you still have the deep water. So they would still have similar ecosystems that would probably be occupied by similar species of sharks. That's really all I have to say.
Well, the ecosystems would be, so when God originally created animals in perfect working order, in fully working ecosystems, that was all destroyed at the flood. And then after the flood, the repopulating or reemerging of these species after the flood in different environmental conditions, right? It'd be a totally different environment. These populations that would recover and that would vary from the original archetypes, they'd be going into different parts of the world that would be totally different than what was in the pre-flood world. I mean, God created a perfect world, and he would have put these specific populations of animals in their respective in communities in their respective ecosystems and locations on the planet. I mean, what we see today in terms of where an animal group is, we can't assume that's how they would have been or where they would have been in the pre-flood world. I don't know. Jackson, I want to give you a chance to respond to that, and then we're going to let Jungle Jargon and Alec Cox give their input on this as well. So, Jackson, you go ahead, take as much time as you want. If you disagree with what I said there, point it out, and then we'll let these gentlemen jump in.
That's all right. We can let Jungle Jargon jump in.
Yeah, there's a couple of things that we need to see. That, from what I understand, is that everything basically was liquefied. All the sediments were liquefied and picked up, and then redeposited. And the other thing we need to understand is that the lowest parts, like you're suggesting, the lowest parts were in the sea or the ocean, what we call the ocean, which doesn't have sediment layers. So all those sediment layers were subducted. They went into the magma or whatever it was and melted. So a lot of the evidence is gone. And the sediments that were redeposited the way we see them. But everything down to the unconformity was scraped up. Everything below the unconformity is sediment layers from the creation week water world. If you know what I'm referring to, there was a water world. I mean, the whole world was covered by water on the first day before dry land appeared. So those sediment layers from below the unconformity, those are layers from the water world before the creation event, before the animals were created, basically. And the only fossils in those sediment layers are only in the uppermost layers, and they're only, small fossils that are, not vertebrates or anything. But a lot of like what you say, the shark teeth, everything that was supposed to fall to the bottom is the ocean, and the ocean was all subducted, and what was redeposited, everything was just liquefied, scooped up and redeposited, and that's the order that we see. That's how it was redeposited on land. But that's not the deepest part. The deepest part is in the ocean, which was subducted and went into the magma.
Okay.
Yeah. I also wanted to add, Donnie, you touched on it, the fountains from the deep. So we still know that, through findings of seismic and other geological instruments that they've used, that there's also some sort of an ice core thing that's in the mantle, hasn't melted yet. There's also water, under the crust. So you have the crust, then you have the mantle. So there's water still under there. But the fountains of the deep would have been the majority of the water that came out, not from the 40 days of nights of rain, but the water that came out from under the earth, which would have totally destroyed the creation of Genesis 1. Okay, so what we have now is not what Genesis 1 was. It's totally different. Now, I used to kind of believe in the Pangea. I believe in a supercontinent, but I don't believe that just because Africa and South America match up, and all that stuff. I think that's just because, if you did drain the, because they have satellite pictures where you can take the water out, of the way, and everybody has a continental shelf, and then you have some sort of a deep chasm. You have mountains that are under the ocean that are taller than what we have on land. Actually, the tallest mountain in the world is Mauna Lea, which is in Hawaii, the Hawaiian Islands, and that's the largest mountain in the world. It goes from the bottom of the seafloor all the way to whatever it is at the height of Mauna Lea. And you have that, which would have killed a lot of animals straight out, the sea creatures and land creatures. So whatever wasn't on the ark would have been, could you imagine? I don't know if you ever seen a water spout. Anybody seen a water spout? A tornado on the water?
Not in person.
Yeah.
Okay,
Yeah.
I have, and you don't want to be near that thing, anywhere close to it. But yeah, it picks up fish and throws them out on the land. So that, I'm not saying that might have happened during the during the flood, but could it? I don't know. Obviously, there was an atmospheric change, which obviously after the flood, the ages of man, obviously decreased, slowly but surely. So there's an atmospheric difference. I'm not going to go into all the oxygen levels and all that. I'm just going to say that there was a definite change. And, the waters from the deep were the majority of the waters. So were there any mountains? No. So if you take, the plate tectonics involved in the crust of the earth cracking open and then starting the, continental drift, if you will, at a more rapid speed than it is today. And then you take that and then you put it in the equation of the, the rains that would have also been, coming down for only 40 days, 40 nights. So a month of rain, you get that in Central America. And, the basis of the fountains of the deep just changed the whole Earth's thing. So when you have fossils that you find that jungle jargon was talking about in these higher layers, it was all, I hate to use the jar of clay and all that stuff, with sand and everything, but it does seem feasible that that's exactly what happened and why the ark was built, so that they would be on top of the water, which was higher than what was going on underneath. So they were kept in there safe while that was going on. And I mean, we don't know exactly what happened. This is a hypothesis, but just judging by the, fountains of the deep. Okay, I've seen geysers. I've seen, many different things. Water is very, very powerful. So if it's under extreme pressure, it can cut through anything. Except a diamond, I guess. Maybe they've tried it, but I don't know. But the fact is all that pressure of water coming up at once, quintillion gallons of water, just flowing out. And then, all of a sudden you have this fossil record that they have the index fossils. So, how they judge the fossil is what rock it's in, what rock is by the fossil. Because, we find these fossils in this layer, but you also find that fossil in other layers. Now, I can't pick out the one, but trilobites come to the worm, the sea worms that they find in the Grand Canyon. That comes to mind. They're in different layers. They're not just one layer. So some sea creatures could have lived a little bit longer than others, maybe the burrowing ones. But the majority of your argument with the shark's teeth, look, I've found many shark's teeth. I don't want to be in shark's teeth, but fact is that shark's teeth, I would love to see if they didn't have the collagen, the problem with their skeletal and everything like that. If one was sitting on top of Mount Everest with its jaws open, like you see at Universal Studios, but that's not going to happen. And that's what everybody has a picture of. It's kind of like, oh, well, how come there's not a dinosaur, or something like that? Well, we do see dinosaurs like that. Apparently, there's a big dig going up in Montana where they're finding days after days, just finding these, new forms and things like that. They're like, whoa, what's this part of? But by the millions, they're finding bones, just in the middle of the United States. It's crazy. So that's the end of my thing. But I'm going with the waters of the deep just being the bigger disruptor, causing the whole continental drift and what we still see today with earthquakes and things like that.
Well, those are some great points, Alec. And to support that, which I'll do in 1 second, Jackson, take the floor before I give my input and make any points you'd like to, any pushback you'd like to. Go ahead. What are your thoughts?
Well, you mentioned trilobites. Like, there are specific species of trilobites in specific layers, and sometimes even only part of the layer. Like, what is it? Paradoxides, I think it's called. It's only in the middle Cambrian, and nowhere else. Small or large, they're all in the middle Cambrian. So, I understand what he's saying, but I think that's hard to explain.
Well,
yeah,
okay,
let me just add this.
Yeah.
Very, very hard to explain, because if the Cambrian goes across the the whole Earth, then you should be able to find that index fossil in every bit of that. And they probably don't. Probably find it in one site, and a dig takes forever. It's not like you're there for a day and you're out there making sandcastles. It's literally, these people dedicate years and years and years and years just to find one specific thing that they're looking for to make that an index. Because if you find one, you must find multiples. You can't just find one, right? So then you got to find multiples. And then sometimes, by mistake, somebody, takes their little pick and it breaks the whole chunk of rock off and they lose it. Now you've destroyed your evidence. You know what I'm saying, Jackson? So what I'm trying to say is that the Cambrian supposedly is everywhere on planet Earth, right? So if we on land anyway. So if we were to say that this particular species of trilobite is found only in this specific depth of the Cambrian or the next layer up, right? Then okay, that's an index fossil. But you also find other fossils in those layers. But then they throw that up and say, no, there's more of these, so we're going to index this layer with that particular fossil. But that's changed over the years. If you I had to look at it. It's not just the Cambrian, but the next one up. They've changed that layer from the index of certain things because they find a mixture of marine animals with land animals in it. So it's like, oh, how'd this happen? And you can't just go around with an earth mover and just, decide to plow through a side of a mountain and say, oh, let's have a look at this. Number one, the federal government probably won't let you, and all this other paperwork you got to get through. But on the other side, it's what they're finding and then placing in, then it becomes gospel for the evolution side. Whereas instead of saying, hey, we found this, we haven't, fully studied it yet, we're going to print this paper and this is fact. But then they find it and then they erase it and then everything, but it still is in the 14th edition before they get to the 20th edition of the textbook. It's still in there. So you still have 7 years of classes that are learning this stuff, and then they find out, oh, that's no longer existing. Anyway, that's my little input there.
Go ahead, Jackson.
Let me think. Well, there's like the Wheeler Formation in Utah of the Cambrian, where anyone can just go and dig and find trilobites or anything. Same for the Burgess Shale, I think, in Canada. So that's really all I have to say.
Well, here's the thing, Jackson. When it comes to our model, because what you're probably thinking, and you're too nice to say is, well, this whole idea of the fountains of the Great Deep breaking open and catastrophic plate tectonics taking place, this is all post hoc ad hoc to explain the ordering of the fossils and the layers and so on and so forth. For one, I want to say, before we get into the prediction that was made based on this, if this is true, this catastrophic plate tectonics, meters per second movements through the plates, have to be the case anyways to explain the geological features of the Earth, Jackson, because present processes such as the snail-paced movements through the plates that we see, right? The plates move like what? An inch a year or something. They can see through satellites, that type of movement, those processes can explain the geological features of the Earth, like these massive mountain ranges. It would be like, comparing two cars coming at each other at, let's say, 1 kilometer an hour. Okay. Once they hit each other, they're just going to kind of bump and then kind of stay there and maybe bounce. There's not going to be any like buckling or anything catastrophic going on there. Okay. But if you had two cars coming at each other at, let's say, 120 kilometers an hour, well, what are you going to get? You're going to get buckling. The cars are going to crash into each other. They're going to go up, whatever. You're going to get pieces flying everywhere. It's the same thing with the uplifting and the necessary buckling of the rock strata to create these mountains. If you have snail-paced movements of the plates, that's not going to do anything in terms of uplifting these massive mountain ranges that we see. Okay, take the Himalayas, for example, Mount Everest, for example, as well. But no, if you have catastrophic plate tectonics, meters per second movements of the plates, then you have the necessary amount of force and collision power to rapidly buckle the rock strata to uplift for these mountains, okay? And a prediction that was made, and I got it up on screen here, and I've been asking this. Actually, this is a problem for the most militant evolutionists, because we've pointed out, okay, you think there's a heat problem, and yet we have a confirmed prediction showing these massive slabs of cool rock at the base of the mantle where they should either be warmed up or melted. If there was a heat problem, like you're pointing out, why aren't these cold slabs completely melted? Because remember, it's a confirmed prediction of CPT. So it was John Baumgartner. I'll go through this quickly. The worldwide flood, Baumgartner realized, provided just the conditions, okay, for what we see in terms of the plate movements. He proposed that the original pre-flood supercontinent, which God made in Genesis 1, was surrounded by cold ocean floor rocks. Beneath these were the warmer, remember how I was talking about earlier, how you'd have the cold ocean floor as the pre-flood world, but that would be kind of recycled or replaced by the warmer mantle rocks during the flood. Well, here we go. Beneath these were the warmer mantle rocks. Since heat rises and cold rocks sink, the ocean floor was just waiting for a trigger to begin sinking into the mantle. The trigger came at the start of the flood, he believed, when a massive crack opened in the ocean floor as the fountains of the Great Deep were broken up. So you're thinking, oh, starting point, the Bible, right, Jackson? The Bible can't tell us anything, whatever. It's not accurate in history. This is post hoc ad hoc. Okay. So Dr. Baumgartner made a prediction. If this is true, If this is true, then we should see X, Y, Z. Testable prediction, right? So here we go. Once this crack opened and the ocean floor began to sink, the plates moved rapidly until all the cold material had sunk into the mountain, the pre-flood ocean crust. Okay, the pre-flood crust. Using a computer simulation he developed for his PhD thesis research, Baumgartner proved that this continental express could have happened. His model led him to predict two discoveries. And I've been using this argument now for like 3 years in debates. And still no rebuttal. I'd be curious to your rebuttal after when we're done, Jackson. Yeah. Unlike the situation in the present, mantle material during the flood circulated from the bottom to the top of the mantle and back again, because the cold pre-flood ocean floor sank only about 4,500 years ago. Notice this, Jackson. And it would take many millions of years to melt colder material, right? So if what we're saying is true, okay, when it comes to the pre-flood ocean floor and crust, rapid plate tectonics from the starting position of the Bible, supercontinent followed by fountains of the great deep breaking open, colder material should be sitting at the base of the mantle. Think of it like an ice cube in your coffee. It'd still be there after a few seconds, but gone hours later. So listen, if this material was taken down, okay, to the base of the mantle, Jackson, over millions of years of slow snail-pace movements of the plates. Just like the ice cube in your, if you put an ice cube in your coffee right now and come back tomorrow, it's done, it's melted. But if you went for a bathroom break and came back, it's still going to be there, which means the ice cube wasn't there that long, okay? So if this material was taken down there millions of years ago through slow plate movements, then guess what? It should be either just completely melted or, for sake of argument, just to give you the benefit of the doubt, at least warmed up to the surrounding materials. Okay. But for our model, if they were taken down there just 4,500 years ago from catastrophic plate tectonics, they should still be cold. Just like the ice cube should still be there and be slightly cold if we just put it in the hot coffee. Well, notice this. Not long after, in 1987, geologists discovered evidence that supports both conclusions. Although the mantle is very hot, up to 7,200 degrees Fahrenheit, geologists found what? Slabs of material at the bottom of the mantle that are cooler than the surrounding rocks by as much as 5,400 degrees Fahrenheit. This discovery presents two mountainous puzzles for evolutionary geologists. So this is what I want you to address, Jackson. And if it's the first time, like if you haven't really looked into it, that's fine. Look into it a little bit later. But here's the problems. First, the 420-mile deep barrier seems to prevent plates from getting down to the bottom of the mantle. Second, even if plates could push through the barrier at their present rate of 1 to 2 inches per year, they would melt and match the rest of the mantle's temperature. But the findings fit nicely with Baumgartner's catastrophic flood model. So in a nutshell, what are your thoughts on that? But how do you explain not only the evidence, but the fact that it's a confirmed prediction? Dr. Baumgartner made the prediction based on the biblical starting point and based on catastrophic plate tectonics. The discovery was made that confirms CPT and contradicts uniformitarianism, plate tectonics. What are your thoughts, Jackson?
Well, about the cold material and the core, I don't know. I haven't looked into that really at all. But you were saying that mountains couldn't be formed by slow processes. Well, they could be. There was a lot of force behind them that just kept pushing. You know, it doesn't all have to happen at once. It happens slowly, but the same amount of force, just keep applying it. It just slowly rises up. But yeah, I'll have to look more into the cool material in the mantle.
Well, so if two cars are coming at each other at 1 kilometer an hour and they hit each other, if let's say there's a driver and they're both pushing at the gas pedal, but only at 1 kilometer an hour, are they just going to kind of keep hitting each other like this? Or are they eventually going like, is there going to be enough force at that 1 kilometer an hour for or are they just going to kind of keep like bumper cars? They're just going to keep bumping each other? Like what's going to happen?
Well, I think you need a lot of mass behind it also.
Okay, so if you have two cars coming at each other 120 kilometers an hour, what's going to happen? Now we got enough force, right? Now we got enough power collision. But here's the thing. Okay, I understand. Yeah.
Oh, sorry.
I understand what you're saying, but the CPT model provides the perfect amount of mass, the perfect amount of force to explain the necessary buckling of the rock strata to uplift to form a mountain. But not only that, we have the cold material. Okay. And this is another thing, like the functional ERVs, Jackson. I want you to look into it because, you know, a lot of people are like, well, SFT is doing like a gotcha moment. I've been using a lot of these same arguments for 3 years in debates. I mean, I brought that my first ever formal debate was on Modern Day Debate 3 years ago with R.J. Downard, James Downard, militant. I use that argument. To this day, he has no explanation. They don't have any explanation, actually, for the cold material. I mean, it should be either, I would say it should be melted.
Well, here's an
it's there.
I can think of.
What's that, Jackson?
Here's something interesting I can think of.
Yeah, go ahead.
There's I forget what the layer is called, the sun. Above it, it's hotter than below. And it's something that's hard to explain, and no one has an explanation for it yet. And as far as I can remember.
Where's this?
What?
Where is this?
The sun,
he said. But what Donny's saying is it's only in the area of subduction where the plate went underneath the continent. That's the cold slab that he's talking about.
Yeah, I'll have to look into it more.
But I was wondering what you thought about the evolutionary claim that the order of the deposition of sediments is like according to age, but the point of the coelacanth is just to show that it's not necessarily the correct order, because they found the coelacanth in a certain layer, and then it wasn't there, and they assumed, according to evolutionary theory, that it no longer existed, because if it existed, it should be in subsequent layers. And it turns out that it's still living today. And there's probably a whole lot of examples like that. But the point is that the redeposition of the sediment layers isn't necessarily, or isn't indicative of different ages, especially considering the fact that they're all consecutive layers with no soil in between, no erosion between the layers, mega sequence after mega sequence on top of each other, 5-6 mega sequences, without any disturbances between the layers. And so that order of redeposition, which is a redeposition of sediments, can't possibly be indicative of different ages. What do you think about that?
Yeah, I'm losing my train of thought. I'm getting tired.
Oh yeah. Well, I just woke up, so I'm good to go.
Yeah.
No, but maybe the way I said it was confusing. It's just that the redeposition of the sediment layers, to me, is not indicative of different ages, because they're just consecutive redeposition of the sediment layers that were already there.
Yeah, I was going to say something about the coelacanth. They find it in the Devonian, and then they find it kind of in the middle in the Cretaceous, and then today also. So they recently discovered a coelacanth in the Cretaceous of Texas, I think. I want to say.
Yeah, but the assumption was it was an indication of age, and that it would no longer exist, because, and that's another thing, is that we don't know what's in all the layer. We don't know what's all there to begin with, and they keep finding things, and then say, oh, well, it's in this layer too, or that layer. Then how are you going to know which layer is which from one geographic location to another? Just because there's an indicator fossil in that layer doesn't necessarily mean it's the same layer. It's just, but they make that assumption.
Yeah, that's why I'm saying if something like shark teeth are ever found in the Cambrian, that would really I'm not saying they're definitely not. I mean, I guess they could be.
Right.
If they are, that's a big problem.
And but I don't even have to find shark teeth in the Cambrian to know that the sediments are, consecutive water catastrophic deposition layers. One on top of, there's mega sequence on top of mega sequence. There's a whole staircase, the grand staircase of sediment layers above the Grand Canyon. It's hard to explain. I don't know how anybody could ever explain that through normal processes. That to me, that the whole thing is a catastrophic deposition from the bottom of the Grand Canyon all the way to the top of this grand staircase.
Yeah. Is Donny getting coffee?
Probably. You gotta get his fix. It's all good.
Yeah, I need to get off here in a minute. I'm tired. It's about 4 o'clock.
Yeah, you're a machine, Jackson. I gotta tell you, you gotta be soaking up all this knowledge, but I know how it gets when you get tired.
Yeah.
Fully agree with that.
Yeah, we're gonna get Jackson converted.
We are. It's gonna happen.
Maybe someday.
Well, I did. Hey, Jackson, is it all right if I that little tidbit about the Pascal's wager thing about you going to death with your evolution?
Well, I think you can believe in evolution and Christianity. I don't think you have to have either or.
Yeah, see, that puts death before sin, so it's a problem.
Yeah.
But the yeah, so it doesn't agree. So if you don't agree with the first gospel, how can you agree with the gospel of Jesus Christ? First gospel, Genesis 3, what is it, 14 through 16, where basically the seed of the woman's going to crush the serpent's head, and he's going to bruise his heel. So it's still going to hurt the one that's crushing the serpent's head, but it's going to dismantle the serpent.
Yeah.
And so the problem with evolution is you don't have a perfect God that loves us. He just put it out there, and some guy put it in the chat earlier about God just rolled dice until he got snake eyes. It's just like it's just incredible how that's not even a logical way of thinking. So the thing is, I will go to my death preaching Jesus Christ. I firmly know that. I've been there. I don't care who it is. I'll say, hey, how's Jesus Christ treating you? If they look at me funny, and then I go, well, what do you mean? Then I know I have a conversation. If they go, oh, and flippantly say it, I say, well, maybe we can talk about it later. I always give them an option. But it's it's something that when people have pre-assumptions to what they've heard, but they haven't read and they don't understand, because all they've gotten is like snippets from CNN type, how we get our news these days, little gotcha type thing, but they don't read the article. They just read the headlines. You didn't get the full picture, because a lot of times, just like we were talking about in papers, the abstract doesn't match the actual paper or the conclusion. So just let you know that.
Okay.
And there is an actual verse that says that it's better to fall on a rock and be broken than be scattered like dust beneath his feet. So do you know what that's referring to?
Not really.
Well, it's a reference to Jesus it's better to, stumble or be broken with the truth of Jesus than to be scattered like dust beneath his feet when he returns. I mean, that's an actual verse. I'm not making it up. It's not.
Yeah.
It's an illustration.
Yeah. Well, I don't rule out a God. I've said that a lot, but yeah, I'll think about all that.
Yeah.
So, Donny, you still there?
But don't think we have it all together too either. I mean, we're still trying to we're not perfect ourselves. We're trying to get things in order too, and when Jesus returns, then we're going to find out who actually was sanctified, who was, putting to death the deeds of the body and stuff like that, and who was living carnally according to the flesh and all that stuff.
And jungle jargon, you bring up a great point. It's we're all broken vessels, and we're only made perfect through Jesus Christ. There's no way we can be perfect. We don't get a start over. It's not like back in elementary school days where it was it's a do-over, tap tap, whatever. We can't do that in life. Once you mess up, that's it. We don't have to go through the Levitical laws, but at the same time, we are broken creatures, and as Jungle Jargon said, we still struggle. It's not everything's all roses and I'm pooping out rainbows. It's not going to happen that way. So
yeah. Somebody in the side chat says Jackson looks like he's about to hit the floor, and he's not wrong.
Well, let me jump in here, gentlemen. I just wanted to thank Michael Knighton for the super chat. He says, Jackson, have you heard of the Guadalupe skeletons? Jackson, before you answer, after you answer, everybody give their final words. We just hit the 3-hour mark. I got to be up early tomorrow, and then we also have shows here. I think the next 3 or 4 nights, some pretty big debate. So, So Jackson, answer that question. Then we'll allow JJ and Alec. Always a pleasure, brother. Great points, brothers. I should say JJ and Alec and Jackson. Always a fruitful, edifying discussion. Anybody who just got here, Jackson and I had an informal debate discussion on ancestry for the first probably about 2 hours or so. So if you just got here, definitely go back and check that out. Tons of fun, lots of great topics. So it's understandable, Jackson, you're ready for bed, and I'm pretty tired too, ready for bed. So Jackson, answer that question, then we'll get some final words from JJ and Alec, and then we are going to call it a night. So go ahead, Jackson.
All right. Well, I'll say some final words too.
Yeah.
I was supposed to hang out with this girl tonight, so I'm kind of disappointed, but this was a close second, I guess, kind of almost. But no, I haven't heard of the Guadalupe skeletons, so I'll have to look into that. Yeah, I'll do some research into that. So, yeah.
Well, I appreciate it, Jackson. And I think it was meant to be that your date was canceled, but you had a more important debate with young Earth creationists. So
yeah,
I appreciate you being here. JJ, thank you, brother. Alec, thank you for being here, brother. Final words, final thoughts, boys.
Yeah, it's a better appointment, but anyways, I just wanted to mention the three greatest discoveries in physics, geology, and genetics. That's on my channel that I discovered. I just want to put in a plug for that.
Yeah, check out Jungle Jargon's channel. He has some really interesting stuff on the history of origins.
It's
the ancestry is amazing.
Yes, the most recent video right now that I have.
Yeah, you're doing great work, Jungle. And yeah, I want to encourage people to go subscribe to Jungle Jargon. He's doing some great work, especially pertaining to human origins.
And the haplogroups,
yeah. So you're doing great work on the haplogroup. So check out Jungle Jargon. And Alec, final words, brother?
Yeah, well, our Creator God loved us so much that He sent His Son to die on a cross to save our souls, so that whoever believes in Him will not perish but have everlasting life.
Amen. Amen. The best final words that you could say. So, one day, Jackson, I'm just going to have you on, since we've debated the science so many times. We'll just have a discussion on the gospel. I think that'll be good. So
maybe we can do that.
All right. Well, I'm going to let you guys get out of here. I'm going to do a quick outro, and then I'm going to be back here just for 5 minutes, just to go over some announcements, because we scheduled quite a few events. So everybody in the chat, stick around. I'll be back in about 30 seconds as I let these gentlemen out of the show. So blessings, Jackson,
God bless you, Donny,
JJ, and Alec. I appreciate it, Alec. God bless.
God bless.
All right, that concludes another show. Kind of a last-minute decision to do this discussion with Jackson, but as I said, when we first went live, Jackson and I had some disagreements and some debate over Facebook. And since he has the same mindset as myself, we thought, you know what, instead of wasting time in an endless comment war, let's get in here and discuss. We discussed ERVs and human evolution, Australopithecus, Sediba, and some good topics. So definitely check that out. We will be back tomorrow night with George Bond and Professor David McQueen, a couple of great brothers in the Lord and our geology experts on Standing For Truth Ministries. They're going to be here with me tomorrow, fellow SFT team members, and we're going to be discussing order in the fossil record, something that Jackson and I spent some time debating tonight. So that'll be at 9, a little bit later than usual with Professor McQueen, but we will be here at 9 o'clock tomorrow. So make sure you're here for that. We'll have some questions as well. And the first hour and a half, we're going to address order in the fossil record, but then we're going to be talking about ecological zonation. And then the last half an hour, we're going to continue. Professor McQueen's going to continue his response to Gutsit Gibbon on the fossil fuel industry. So if you've not yet seen the previous 2 videos on that, please check that out. I've got them linked in the description box. Then the very next day, we have the much anticipated debate, all confirmed. I've confirmed all of these for this week. Where's the graphic? Here it is. The total inability debate, CJ Cox and Dr. Leighton Flowers. So that'll be the following day. I'm sure there'll be an epic LPP after show for that. Then we're going to have the battle continues. Mark Drysdale and Dr. Dino. They're going to be debating at the end of the week. And I'm sure we'll have an epic after show for that as well. And check the upcoming live stream section, because we've got a ton of debates, interviews. We just scheduled this as well. Dr. Mark Armitage, we had on last week, irrefutable presentation or discussion, I should say, on dinosaur soft tissue and biomaterials. Matt and I spent a great deal of time studying the so-called best criticisms and objections to dinosaur soft tissue, confirming young Earth creation. and presented them to Dr. Mark Armitage, and he dismantled them, decimated them. So please share that around, guys. It was this one here, because we left no stone unturned, and it's 2022 and evolution and deep time, uniformitarianism or actualism, whatever you want to call it. It's dead. It's dead. Is evolution a reasonable scientific theory? This one's going to be fun. Brian Emery and Dr. Ken Hovind. I love the thumbnail here. So I appreciate Brian's sense of humor here. Guys, that's going to be an epic debate as well. Yeah, check the upcoming live stream section. I mean, I think we got like 30 shows there. And I just wanted to, good to see everybody in the chat. Anybody interested in Standing For Truth clothing as well? This is our heterozygous Adam and Eve shirt. We've got tons of cool SFT gear, mugs, shirts, workout clothes, books, so on and so forth. So you can check out our website for that. We have two separate websites. One website, creationistclothing.com, where you can find this stuff. And then our main website, which I am going to go over real quick right now, because a lot of people ask me, and I get it through email and requests as well. I've been getting a lot of requests through the contact form here, guys. So if you want to get a hold of me, you have specific questions, concerns, comments you wanted to make, feedback, you just want to get a hold of me for whatever reason, get a hold of me through there. I get many emails through there a day, so I appreciate it. And one of the common questions is, what's the best way to support the ministry? A lot of people can see that we're growing. We're doing this full time. We have a team right now. And when we're not live streaming, we are either coordinating with each other, studying, preparing for future shows. We've got books on the way. We've got research projects, movies. We're working on Dawkins, debunking Dawkins will be our next YouTube movie, as well as several books we're working on. So we've got, I think now here's some of our best-selling ones up here that I highly recommend. The Independent Origins Handbook, especially. I'm glad to see that one's doing well. We got a ton of books right now. You click the shop now, you'll see them all. These next several books we're working on, especially the one I'm doing on human evolution, I am doing a ton of research for this book. So we're not rushing to get these ones out. We want to do them right. We want to make sure they're irrefutable, well documented, cited. So we're spending a lot of, if we're not live, we're pretty much working day and night for you guys. So we got a team. We've been blessed with Professor David McQueen, who's a retired geologist, university professor. So he's been doing a ton of work and research on the geology side of things. So I've also got a website designer who's up on all of this. We have so much content, so sometimes it gets overwhelming making sure that it's all up on the website. I just did an update last week, so we got our most latest shows up here: Dinosaur Soft Tissue, also Russ Miller. And for people that just want a quick answer to a common question or a common criticism or challenge, actually I should check from my phone the chat, make sure everything's coming through, guys. Give me an A-OK on the audio. Okay, I appreciate it. Yeah, Sea Science. Yeah, I'm looking forward to that as well. Greenie. Yeah, Greenie's got it. So, actually, before I go into the Patreon, answering common questions and refuting arguments. So if you just want a quick answer to the common questions, we've got a little bit of everything for you here. From limestone to dinosaurs to lies in the textbooks to nested hierarchies, DNA, down to is the New Testament reliable? Tons of stuff. I mean, this website is really what I visioned, what I dreamed of when I first started. I wanted an all-comprehensive website that just destroyed the critics. And I think that's what this is. So I really found a solid website designer for this. But yeah, so a lot of people ask, how can we best support you guys? What's the best way? Especially when it comes to like first-time donors, you can do it right through the donate box. It goes right to PayPal. That's always much appreciated. But you can become a patron. And that's one thing I haven't been, I've been active on it, but of late, not as active as I want to be because there's only so much time in the day. But we do have a goal now for 2022. We want to hit 100 patrons. Okay. So, you know, we need your guys' support, of course. It can be as little as, I mean, to be a patron, it can be $1 a month. I think it is. Because we're finding now the more that we've grown, the more projects we have, our production value, my new studio here, you know, you could probably tell, you know, we got a 4K webcam going on, and we want to expand into the community as well. You know, we'd love to, I'm in Canada, but other members of Team Standing for Truth are in the States and Australia. But we'd love to get out into the community, and we want to get a really good camera for that, where we can start doing our own seminars and presentations and lectures out in the community. Get those filmed for you. Conferences, you know, hopefully we can meet up to do that. I mean, we just have endless ideas. So the more that we find we grow, we want to expand things out. The more events we do, the more bigger names we get on, the more expenses we have. I mean, you know, it's the truth. We're doing this full time, but yeah, the more we grow, the more we do, we just find that it's the more the more we expand and put in, right? The more expenses that there are. And then we've got a ton of but we're pretty much, I mean, I don't know what channel puts out more content than I mean, we got a ton of content, and already I think March is pretty much booked completely. We got April and May going right now, but because of all the behind the scenes work, so you know, we had a discussion, and yeah, we think being more active on Patreon. So if you're new to the channel, but you're especially looking for a good way to kind of support us, you know, we won't let you down. We're working day and night, and we want to put out the best content. We want to have the best guests, and we really have some great ideas, and we are motivated to expand big time. We had 2 conferences last year online. We'd love to do an in-person conference, but we do already have, we're kind of in the starting, we're in the start of organizing our Defending Genesis Conference 2022. So we had one in 2021. We also had the Genesis Flood Week in 2021. So, but we want to make the next conference even longer. We've been doing 3-day conferences. We'd love to get a 5-day conference. Yeah, get even more guests, names, and those we have on, we also like to support as well. So, you know, whatever we get in terms of support, we put it right back into the ministry. And, you know, you guys are the life and blood of this channel. Without you guys, I don't know where we'd be. You know, we are a listener supported ministry. So yeah, Patreon. It's easy. Like I said, as little as I think $1 a month. So we have a goal right now, 100 patrons in 2022. Once we hit that, maybe we'll get a new goal. But this is the way that we're going to be able to expand. This is the way that we're going to be able to continue putting on all of these shows, these big debates. People love that we've been doing a lot of theology debates as well, and soteriology debates. I have so many requests coming in every single day, and tons of great ideas. You know, I get long requests through email where people are interested in having us refute certain videos. A lot of great recommendations in terms of debates. You know, a lot of people, they'll say, you know, have you ever had this person on or that person? And I probably spend about 3 hours a day on email. I'm the email guy. So I'm kind of OCD when it comes to organizing events and just making sure everything works smoothly and goes smoothly, especially for our Evolution debate challenge right now. So I want to do all of the requests that we're getting and all of the amazing ideas. You know, there's only so much time in the day. I'd like to get to a point where we can, let's say we have a month that's just packed with debates. Hey, we'll have some days where we do 2 debates in the same day, you know? So again, you guys are the life and blood of this channel. And I've had a lot of people ask me, And so I thought I would just do a video on it. But since we had an open mic here and an informal discussion / debate with Jackson, I thought I would just address it now. So again, yeah, our goal hit 100 patrons in 2022. And that way we can feel comfortable knowing that the ministry is running smoothly, the costs are covered, and we can continue expanding and working day and night to bring the best debates, the best interviews, the best movies, the best content, the best open mics, and like I said, the best books, because I promise you guys, these books Matt and I are working on behind the scenes, especially the one on human evolution, it's going to blow your minds. It's going to be a game changer. And the critics who are already running, they are going to be running faster than they've ever run before. So that's kind of all that comes to mind right now. But again, I want to thank everybody for the support. Even something like sharing around the content, hitting that like button, passing around our website, anything like that is a huge help. But yeah, we got some ideas, and we got some goals, and I think 2022 is gonna be a big year. But the big thing is we're really finding now that the more we grow, the more we expand, the more expenses that accumulate. So yeah, if you want to support us, we greatly appreciate it. We won't let you down. And Patreon's a great way. Patreon's a great way because, like I said, as little as $1 a month, you can be a patron. And I don't have any of those tiers or anything like that where you gotta, donate this much a month and you get access to. Everyone's on the same tier. You can donate $1 or $100 a month, and you're still going to have access to the content. So yeah, anybody has requests, ideas, again, go to the contact section of the website. Let me know. I've got a list. Of a huge amount of videos that Matt and I will be addressing and refuting. One right now that we're really working on. I've got everything ready for it. We got to find a good day to do it. It's a video that Gutsy Gibbon put out on, I believe it is the Thinking Atheist channel. We don't really like to just do blind rebuttals. We put a lot of research and effort into the response videos that we take on. So the massive list of videos here, I'm just going to the channel section. We want to get to them all, because we want to refute all the biggest, baddest videos out there. We want to expose all of the weak arguments. So we like to do a lot of research, prep your slides, prep a really good show for you. And you guys know that from our response videos we've done to people like Vice Rhino, Professor Dave, Simon Dan, Aaron Raw, Dan Stern Cardinal, Pelagia. I mean, those videos, they go untouched. The critics run. Anyone who just got here. So here's an example of the upcoming live stream section. So we got a ton. We're ambitious, we're encouraged, we're motivated. We want to be the ministry that's putting out the most content for you guys. We want you guys to know there's going to be something nearly every day for you. and not just anything. I mean, really fun, edifying events. And this Evolution Challenge series that we've been doing has been a ton of fun. Epic showdown right here. Ken Hovind and Snake was right. They're going to be debating again. But yeah, for example, here, I guess you can't see it because I'm live, but usually it shows up as the Simon Dan video that we've done response to his video. So he put out a new video recently. I believe he failed miserably, trying to refute one of our videos with Dr. Dan Biddle from Genesis Apologetics on how man lived to 900. And it was a flop. But yeah, we want to get to that one. And we want to glorify God and defend the truth of biblical creation. So anyways, that's kind of all that comes to mind. We've been going for 3.5 hours now. And if you just got here, check out the whole show. We had a ton of fun. Jackson Roe and I had an impromptu, informal debate, and we touched on a number of interesting topics, especially endogenous retroviruses was very thorough. That's what we were, see, and here's the thing. We need more evolutionists that are like Jackson Roe. You have no idea how many keyboard warrior atheists there are that comment on Facebook, that comment on the YouTube channel every day. Beamsy is a prime example. Beamsy, he was talking smack for, I think, months, calling me a liar, saying I misrepresent the data. And finally, he had the hair on his legs to join some of our after shows, and he really got exposed. I mean, he had no real rebuttal to genetic degeneration and genetic entropy, and he pretty well had to admit defeat. But that's one thing I love about Jackson Roe, is he's the kind of guy where you're arguing a little bit back and forth on Facebook. Okay, we were arguing about ERVs, and we just both agreed, let's take this online. Let's make a thing of this, you know. So if only there were more evolutionists who would graduate from keyboard warrior status in mama's basement and would take the evolution challenge, because it's in these open mic discussions, it's in these live formal or informal debates where you can really press the evolutionist and really expose the massive amount of storytelling and imagination that takes place when it comes to pond scum, to people evolution. I mean, I've been asking now for 3 years in formal debates. I'm up to almost 100 formal debates, and I've debated extensively the topic of endogenous retroviruses. And I've been asking over and over again, show me one example of a non-functional endogenous retrovirus going from non-functional to something incredibly functional in determining cell types, regulating genes, functional in the embryo, okay, functional. There's a certain class of retrotransposons, and it's hilarious. I debated RJ Downard, I think, 4 times, but my last debate with him was a formal debate on human evolution versus Adam and Eve. I mean, I don't think he provided even one line of evidence for human evolution. But there's a certain class of retrotransposons in mice that if you snip it out, okay, the mouse is developing, right? In the embryo, it's developing. You snip out this certain class of retrotransposon, and you know what happens? The mouse dies. It dies suddenly. The developmental process stops. It ceases. And you know why? It's because the mouse, the development of the mouse, the embryological development of the mouse depends on that retrotransposon. And you snip it out and it dies. So apparently mice were just not having kids for millions of years until they were infected with viral DNA, and eventually, you know, the viral-like element, you know, adopted a new function in the embryo, and now, you know, mice could start having babies. I mean, this is how ridiculous it is. And I asked RJ Downard that question, and he was just silent. I mean, he was really lacking an argument. So, you know, I debated a biologist on T-Jump's channel, Tom Jump, who's going to be here debating Kent. And I asked him, I said, show me a paper of a non-functional ERV going from non-functional to functional. And he said, if that paper existed, I'd show you. Meaning it doesn't exist. It's a lot of imagination. Just like we pointed out with Jackson Roe tonight, time is the hero of the story. Time and imagination is the god. The gods, imagination and time for the evolutionist. So yeah, right here, this fascinating paper. Long disregarded as junk DNA or genomic dark matter, endogenous retroviruses have turned out to represent important components of the antiviral immune response. These remnants of once infectious retroviruses not only regulate cellular immune activation. Oh, it was co-opted. It was co-op. When they say co-opted or co-option, you know what? That is mere uninformative gloss. It's storytelling. It's imagination. It's void of any real empirical scientific observation and evidence. It's a rescue device. The best lines of evidence that the evolutionists can point to have been overturned. And they're actually in favor of the separate ancestry model. Pseudogenes overturned. Chromosome 2 fusion overturned. Endogenous retroviruses overturned. Allu sequences overturned. Nested hierarchical patterns in morphology, in anatomy, genetics overturned. Nested hierarchical patterns in mitochondrial DNA overturned. Okay. Shared gene order along chromosomes overturned. The junk DNA paradigm overturned. And it's amazing because from our starting point of separate ancestry and design diversity, we would predict, as I pointed out, function. And that's exactly what we find. One focus will be on recent advances in the role of ERVs as regulators of antiviral gene expression. Isn't that amazing? Junk no longer. ERVs are integral and important components of immune responses. I mean, it's a great time to be a biblical creationist. So, Jackson and I discussed in great detail Australopithecus, Sediba. Yeah, we had a lot of fun. It was a lot of fun. So, I think that's it for now, guys. And I'll be debating Luca Medugno actually soon on human evolution. I'm looking forward to that. Luca's a great guy, and I appreciate him being a good interlocutor, and he prepares for these debates too. So, anyways, I guess no evolutionists are going to join to defend their religion of pawns come to people evolution. We're going on 3.5 hours, guys. Again, we'll be back tomorrow. And if you want to support us, we greatly appreciate it. You got Patreon. You can do it through our website. You guys are the life and blood of this channel. So God bless you. Thanks for tuning in. And we will see you tomorrow night for a show with George Bond, Professor David McQueen, two Standing for Truth superstars. We are going to be discussing order in the fossil record. God bless all. Standing for Truth is out.